I'm standing here, I'm looking around, and I'm wondering what made nearly 300 scientists and other participants leave their daily activities for 2 days and come here. I suppose you may be aware of something that also Charles Darwin knew. Let me quote Charles Darwin for the beginning. It is the long history of humankind and animal kind as well, that those who learned to collaborate and improvised most effectively have prevailed. For me, it sounds like the aim of this conference. Ladies and gentlemen, I would like to warmly welcome you, all of you that are present here today with us, and all of you watching us online, at the 4th Interdisciplinary FNP Conference. My name is Katarzyna Pronobis. I work at the Foundation for Polish Science, and it's my great pleasure to host this meeting today. I have already revealed you the main aim of the conference, so let me now tell you what we have prepared so you can meet, exchange, establish inspiring scientific contacts. We have designed thematic panel discussions, presentation sessions, and meetings at your scientific posters. And if you have ever wondered where and how to apply for prestigious grants, you have the one and only opportunity here, because tomorrow our colleague representing Polish Science Contact Agency Polska, in short, of Polish Academy of Sciences in Brussels, is going to tell us about the granting opportunities available within the EU funds. Moreover, today and tomorrow, our colleagues from Excellence in Science Department of Polish Academy of Sciences are also here with us. They are here to inform you on how they can support you in applying and implementing various grants, including ERC grants. You will find them in the posters area during the breaks. And if you want to have all important information in one place accessible, please go to Apple Store or Google Play and download the conference application. What you need to type into the search engine is FNP Conference. As for the Wi-Fi, the login and the password are at the back of your IDs. Last but not least, due to the fact that our conference is co-financed by the EU funds, if you have not signed the attendance list yet, please do so. You will find it at the reception desk. And now, let the 4th Interdisciplinary FNP Conference begin. So, ladies and gentlemen, I'd like to warmly welcome on stage Professor Maciej Żylicz, the President of the Board of the Foundation for Polish Science.
So I would like to welcome on this conference, Interdisciplinary FNP Conference. This is something which we decided to do a long time ago. Each organization like Wellcome Trust or HHMI or others, they have the conference like this for the awardees, or what we call beneficiaries. And of course, there was a break because of the pandemic, but we promise you that we will have such a conference every year. And the idea of this conference is to build collaboration and also networking, and also exchange the information about how to apply the results which were received during the grant work. And this is something which, this application of the results is something which is a problem for all of us, because using money which belongs to everybody, public funds, it is very difficult to achieve the goals. And mostly because the Polish law system is ambiguous. There is a lot of different interpretations of this law. We need to be extremely careful. And actually, this is something which we will discuss today in one of the panel. And one of the panel in afternoon will be about building startups in academic landscape. And so the scientists, successful scientists which open their own business will discuss with the venture capital representatives, which usually invest in companies which have market-relevant products. And this is the only one way to make an application of your results, but extremely, it is important. So please come to this panel discussion and ask tough questions. I promise I will also ask questions about this. There are also other panels which are extremely important for us. Tomorrow in the morning, we will discuss the system of financing science in Poland, which is, I would say, far from supporting excellence in science, and unfortunately, and is not supporting finance implementations. This is something which, again, will be discussed on such a panel. Another panel will be about people who actually working in our grant system, received money from us, and received the ERC grant. And this is something which is still, I would say, very unusual. It should be more often happened, but it doesn't. So again, we will discuss why those people were successful and how we should focus on some issues to receive the ERC grant system. I was for 6 years ahead of the ERC Advanced LS1 grant system in Brussels, and we received only one application from Poland. And unfortunately, even this one was not even go through to the second step. So this was I was very frustrated, and we will discuss this. Another panel will be about those who have already FNP grants, which was supported by Smart Grow Operation Program, but and received extra money to fight with the pandemic. So again, it will be very interesting to listen what they accomplished and what they did for this actually extra money which they received. And finally, at the end of our meeting, Michał Pietras, program director in the Foundation for Polish Science, will talk about the new EU FNP program, which will start hopefully next year, in the beginning of next year. We are already delayed for 2 years, but I cross my fingers that this program will start in the beginning of next year. So once again, I would like to welcome all of you in this meeting, and the real goal of this meeting is creation scientific networks, initiate cooperation, and exchange information of the implementation, this success and also failures in this, because sometimes the failures give you the more information how next time you can be successful. So thank you very much for your attention, and I invite you to actively participate in this conference. Thank you.
Thank you, Professor Żlic, for the welcoming words and all useful information. So the event has been officially opened. Now it's time for the first presentation session, Research Results Based on International Cooperation. By this occasion, I would like to thank all members of the Scientific Selection Committee of this conference for their work on choosing the projects to be presented on this session. This session is going to be moderated by Professor Michał Zawada. Professor Michał Zawada from Nicolaus Copernicus University in Toruń, Team Program Laureate, and member of the Scientific Selection Committee of this conference. Professor Zawada, the floor is yours.
So good morning. First, I'd like to kindly invite to the scene Professor Bożena Kamińska from Niecki Institute at Warsaw, Adrian Kobiela from University of Gdańsk, Dr. Marcin Kuźniak from AstroCent, and Dr. Radek Łapkiewicz from University of Warsaw. Please just sit for a moment. As we all know, international cooperation is a condition sine non qua for Foundation for Polish Science to getting any grant. Foundation always treated really seriously. Probably that's why the first panel is devoted to projects that not only provide excellence in science, but successfully make international cooperations. I would start with inviting for the first talk of Professor Bożena Kamińska, the head of Laboratory of Molecular Neurobiology at Niecki Institute, the director of Postgraduate School of Molecular Medicine in Medical University of Warsaw, elected member of Polish Academy of Science and EMBO, and also professor received a prestigious award of Foundation of Polish Science.
Thank you very much for allowing me to discuss our project. Let's start with the presentation of the project. So we've got a TeamTech Core Facility Award, and I would like to show the logo of the project. If I can we run the presentation? Okay, so our project is the project that we wanted to create an NGS platform for comprehensive diagnostic and personalized therapy in oncology, and I will briefly tell you what about this project is. So I like very much the quote by clinician Sir William Osler, who said that if it were not for the great variability among individuals, medicine might be as well a science, not an art. And we right now know that the single-cell nucleotide polymorphism, which is a spontaneous process of single-change base, is actually a major source of variation between each of us. And when it occurs in very important place of the gene, it makes also a disturbance in cell function, and it leads to the disease. And the biggest effort of recent clinicians and genetics show that this is really a base for the pathogenic mechanism occurring in many diseases, including tumors. So we focus on the neuro-oncology, first of all, because malignant brain tumors called gliomas are the most frequent and aggressive, and there is no effective treatment. So we thought that if we learn a bit more about the genetic changes, we can find vulnerabilities which were not previously discussed, and we can change the prognosis and outcome for this patient. So we built a team with Bartek Wojtaś, Agnieszka Kaczmarczyk, and 4 PhD students. And the main idea was that we would collect the data, collect the samples, made the analysis. Researcher would It doesn't work. The laser doesn't work. Researcher would analyze the data, and we will produce the important information, allowing us to prepare a diagnostic test or maybe find a new way of for the treatment of cancer. What we achieved was building a huge network. And I will come to the international collaboration later, but I would like to say that we achieved creating a network of clinicians from different places in Poland. We organized the largest biobank of brain tumor samples in Central Europe with many samples sequenced DNA, RNA. We implemented methods of sequencing and bioinformatic analysis, and these methods were applied, for example, to produce a first comprehensive landscape of human gliomas, in which we show the very integrative view of disturbances which occur in brain tumors, and we found new pathological mechanisms. We also produced the quick test to evaluate some vulnerabilities to chemotherapeutics, and design panel allowing us to diagnose and containing genes which were actionable, which means that they could direct the therapy of the tumor. We also created an NGS platform which was prepared for people to awake public awareness regarding biology, diagnostic, and therapy. And our team created the sequencing core facility. As I mentioned, our achievements would not be possible without international collaboration, and I'm going to show you 3 examples when this was really essential. The first story started with patients. We found the patients, very young men diagnosed with glioblastoma, which is really amazing tumor composed of 2 different tumors. We found the actionable mutation in the VRAF gene, which was supposed to lead this patient to the therapy. The patient unfortunately died because before the formal things were arranged, but we became interested in this. But the tumor was very rare, so we had only 3 samples, and I was looking for the collaboration, and I was really happy to meet Professor Christel Herold-Mende from University of Heidelberg, who was providing us with information, with interpretation, and with samples. She organized for us 7 more samples, allowing us to find that these tumors have unusual combination of some mutation, making them vulnerable for drugs. And we found also a new marker which could be used for rapid detection and diagnosis of this tumor. We published this not very well, but we wanted to publish this fast. We had this idea that this data would be very important for people. The second story is about our biggest analysis, in which we identified the genes which were particularly mutated in Polish cohort. Polish people were not involved in most of the studies, so this was first identification of specific changes in Polish population, and we focus on 2 genes, RECL4 and TOP2A, encoding helicase and topoisomerase, 2 very important genes. And we found this recurrent mutation only in Polish samples. So we thought that it would be something which arises in Polish population. And the reviewers didn't like this idea, didn't believe this idea. So they asked us to go to another database, so-called Glioma Longitudinal Analysis Consortium, of which we were not members. So I asked my colleague, clinician, Dr. Nushmir, to run the analysis, and his student, Natalia Morosini, ran the analysis in 3 weeks, finding no such mutation in another database, which means that we found really Polish founder mutation in very important genes, and the importance is emphasized by the fact that, first of all, as you can see, top mutant patients has really very short survival. The mutation substitution in the protein changes the properties of the proteins, and I'm not going into detail, but when you look here, the first panel, you can see by naked eye that mutant binds stronger and has different properties. The last example is actually international and national cooperation. We found mutations, some recurrent mutation. Multiple dots means that the mutation is recurrent, produces some substitution in the protein, and in collaboration with Dr. Sebastian Glatt and his student, they were actually able to reconstruct the protein structure and show that mutations are in place, which is very important, and then using recombinant protein, they verified that at least one of the mutations, this one, is really killing the activity. So we wanted to see if we can see this not only in vitro or in cells, because we did also cellular studies, we wanted to see in the animals. So I contacted the professor of the developmental genetics at the University of Portsmouth. He was a director of European Xenopus Research Center, Professor Matthew Gooley. And he agreed to work with us. He developed a mutant, Xenopus mutant, frog mutants, which were showing really strong differences in the composition and number of nerves, and also including double mutant had very strong differences in the head development. So we could prove with this collaboration, essential collaboration, that these mutants are really working. So both Hutan Nushmir and Christer Harold Mende were speakers for the meeting I organized in Warsaw. Actually, Christer Harold Mende became a member of the committee, and she invited me also for the consortium Invigorating Immunity Against Brain Metastasis this time, which was selected for funding. So this cooperation, which was started quite early, will be continued, and she also invited me to submit another proposal for hard-to-treat cancers. Thank you.
Thank you very much, Professor, for your really nice presentation. Now we have 5 minutes for questions from the audience. Anybody want to ask a question? Maybe I will ask the first one, which probably is not strongly connected to the topic, but I'm really curious if any new technology that was developed during the COVID pandemic, looking for vaccinations, has any potential to help in prolonging the lifetime of the tumor.
I mean, not exactly, because these technologies related to COVID were targeting COVID mechanism, pathomechanism. So, but maybe the general idea of developing mRNA vaccine, for example, could be very useful, and the idea of having mRNA vaccine against tumor has been in the field for years. Just the problem is that it's very hard to find something which differs tumor from the other tissue, but maybe from the normal tissue. Select the tumor and target specifically tumor cells. But this is definitely a developing issue, and of course, the way of providing vaccine, these liposomes, nanocarrier, which deliver vaccine, are very important for us because we can put any cargo into these liposomes, into these vehicles, and then we can provide specifically, for example, to the brain, because some of the molecules can have capability to pass brain-blood barrier more easily than the regular one. So I believe that some of the achievements would be useful for us.
Okay, just a stupid question. I was very intrigued by your story about this gene that you found in Polish population. Should I understand that this is something specific to Polish population, or just has not been found in other populations because not tested or not known?
I said the mutation is found in the Polish population because the gene encodes very important protein. So it was important to say that this gene could be mutated in cancer. And we found that in Polish population, 4 people, 2 of them from Silesia had the same mutation. So the gene is ubiquitous, but the mutation has been found only in Polish population as a pathogenic mechanism.
Okay, thank you very much.
We didn't see, it's not, and we exclude the fact that it could be germline mutation, which means something passed through generation. Yeah. No, this is a new mutation which was found in Polish population.
Okay, thank you very much, very, very interesting.
Thank you so much. Nice talk. I want to ask I got your good side of this international collaboration, but did you experience some difficulty, maybe based on the bureaucracy or something?
Actually, no. I was surprised because, for example, with Hutan, we just called him, made a Zoom, we explained what we need, and his student was just working for some time. With Crystal, we were working before for another study, so this was, actually you could not send DNA from Germany, but they could send us samples of tumor from Germany. So basically, we didn't experience any formal obstacles in our collaboration. But we knew each other before. I mean, only with Matthew Gill, this was something new. I had to convince him to invest a lot of time and one of the students to go for these mutants, Xenopus mutants. And of course, COVID closed for 2 years almost. Because of COVID, the facility was closed. So we experienced some difficulties, but not because of collaboration.
We have time for one short question. If not, then let's thank again our speaker. And I want to invite now Adrian Kobiela, who graduated from University College of London under supervision of Professor Joseph Kittler. Then he moved as a research assistant to Niecki Institute in Warsaw. And at the moment, Adrian is working towards his PhD in the Laboratory of Experimental and Translational Immunology at the University of Gdańsk, under supervision of Professor Danuta Gutowska-Owsiak.
Thank you so much. So good morning, everybody. And today I would like to share with you a portion of our research on how the epidermis communicates with the immune system. So the epidermis is the outermost layer of the skin tissue. It is composed mainly of cells called keratinocytes, and the top layer of keratinocytes is made up of dead cells, dead keratinocytes, that form an impermeable barrier which protects the deeper layers of the tissue from any environmental irritants. Epidermal filaggrin is a key protein for the formation of this impermeable layer, but it is really a multifunctional protein because it has been found to influence the immune system. It has antimicrobial properties, and what's really interesting in the context of our research is that filaggrin insufficiency, which means that there is abnormally low levels of fully functional filaggrin protein, is associated with atopic dermatitis, as well as other allergic inflammation type disorders, such as asthma, hay fever, or food allergy. So what we use on our research is the keratinocyte cell line, which are called HACAT, and as we aim to study the filaggrin insufficiency, we had 2 versions of this cell line. One is the filaggrin expressing, or SHC, and one is the filaggrin insufficient, or SH filaggrin. Keratinocytes, just like all nucleated cells, produce a variety of different extracellular vesicles. And in our research, we focus especially on exosomes. These vesicles are very interesting, and that is because due to their small size and their flexibility, they can penetrate into the bloodstream or even into the blood-brain barrier. And that's why they can mediate all short and as well as long-distance signaling.
So a very important technical note in terms of general research into exosomes. These vesicles, it's really difficult to obtain pure preparations of these. And this is due to there are always some level present of vesicles of other origin in these exosomal preparations. That's why what we say for greater accuracy, the vesicles that we use for our research, we call exosome-enriched small extracellular vesicles. And this is the nomenclature I'll be using throughout the presentation. So for simplicity, if you remember the SHC keratinocytes, we obtain SHC SCVs from them, and from SH filaggrin hackets, we obtain SH filaggrin SCVs. As there is not still much known about the role of keratinocyte-derived SCVs in the immune system, we aim to investigate how filaggrin insufficiency changes the role of keratinocyte-derived SCVs in T-cell activation in response to antigens. And antigens are compounds that can activate T-cells, but they cannot do it directly because they first need to be captured by the so-called antigen-presenting cells, or APC, and they had to be displayed on the surface of the so-called antigen-presenting molecule. And then this complex only can interact with the T-cell receptor of a T-cell and activate this T-cell. But this can only happen if the T-cell receptor is specific to the antigen that is being presented. In this part of our research, we focused on the lipid antigen presentation, and they can be presented by the CD1A protein. We can divide the lipid antigens into two categories. One are permissive antigens, and these facilitate the binding between CD1A and T-cell receptor, leading to T-cell activation, and the other kind that block this interaction. In this case, we're dealing with non-permissive antigens, and there is no T-cell activation in this case. In our research, we also use the enzyme called phospholipase A2, or PLA2, and that is because it's known to release lipids from biological membranes, such as, for example, cells or potentially SCVs, and these lipids can be CD1A antigens. This enzyme is very relevant in terms of the immune response because it is produced by immune cells or pathogens. It's been found in venoms of different animals, and skin disease has been shown to be enriched in this enzyme. So first, to test the action of our SCVs on the lipid antigen T-cell responses, we designed an experiment with the following setup. We had the antigen-presenting cells which express CD1A on the surface. We added to them our SCVs and the PLA2 enzyme. Then we combined the cells with T-cells, and we performed the T-cell activation assay, the results of which you can see below in the form of spots, and each spot represents a single activated T-cell. In the case of this experiment, we tested two different doses of our SCVs, and what we observed is that while our SHC SCVs were enhancing T-cell response, the SH filaggrin SCVs really did not make any difference. So the easiest way to explain this phenomenon would be that the lipid composition of our two populations of SCVs is different. And this is exactly what we found when we profiled the lipidome of our SCVs. Here, looking specifically at fatty acids, which are one of the lipid species that are present in the biological membranes, we saw that our SHC SCVs had chain lengths of around, the fatty acids in the SHC SCVs had chain lengths of around 20 carbon atoms. And these chains were polyunsaturated, and their molecular weight was just above 300 kilodaltons. In contrast, our SH filaggrin SCVs, the fatty acids of these had shorter chains, which were fully saturated, and they had lower molecular weight. So at this point, we really did see a difference in the lipidome, but how does that relate to our T-cell responses that we are seeing? Well, looking for the answer to the question, we came across a paper which profiled different CD1A antigens by their size and by their molecular weight. And the outcome of the analysis was that the best at eliciting T-cell responses were lipid species that were around 20 carbon atoms in length and weighing around 300 kilodaltons. And this nicely mirrors what we see in terms of the lipid species in our SHC SCVs. So in this experiment, what I've just shown you, you can imagine that this is quite a physiological setup, because you can imagine that in the skin, you would have the antigen-presenting cells, the SCVs, and the PLA2 enzyme all at the same time. However, we really wanted to dissect the role of the SEV-derived lipids in this T-cell activation. And for that reason, we modified the setup of this experiment a little bit. And in this case, what we did, we first digested the SCVs with the PLA2 enzyme, and only then we added them to the antigen-presenting cells. And this ensured that the only PLA2-derived CD1A antigens came from the SCVs, because the exposure of the antigen-presenting cells to this enzyme was minimized. And here, really interestingly, we saw that the SH filaggrin SCVs resulted in a reduction of a response, while SHC SCVs did not make any difference. So again, we went back to our lipidomic results, and As of now, there's not many lipid species present in biological membranes that are known to be clearly permissive or non-permissive, but we performed an extensive literature search, and we related the results of this search to what we saw in terms of the lipidome profile. And what we found that while the SHC-ACVs were decreased in the amounts of permissive ligands, and there was an increase in the non-permissive antigens. And we now also know that the non-permissive antigens bind more strongly to the CD1A than the permissive ones, which is why these are the species that will make a difference in terms of the T-cell response. So we think that this explains the differential T-cell response that we saw. So to sum up our research, we managed to show that the filaggrin insufficiency altered lipid composition of keratinocytes and SCVs. Just for the interest of time, I did not mention the keratinocytes, but if you're really interested, you can see me at poster number 16, which is directly after this session, and I can show you the results there. We saw that the lipid composition differences induce changes in T-cell activity. And what does this mean in terms of the physiology? So we know that the reduced T-cell responses could result in reduced responses to pathogens and affect pathogen clearance and infection. It could also impair wound healing in the skin. But as I said, SCVs can go to the bloodstream and go to different tissues. That's why we think that maybe other tissues like lung or gut could also be affected by this. At the end, I would like to, of course, thank the head of our lab, Professor Danuta Gutowska-Owsiak, our lab members, our collaborators from Gdańsk, Foundation for Polish Science, for funding our research. But I really cannot stress enough how important international cooperation for the development of this project was. We had a chance to work with Professor Sarah Brown, who is an expert on molecular and genetic mechanisms in atopic dermatitis, and that, of course, includes filaggrin mutations. Professor Graham Mock from the University of Oxford, who is really experienced in the field of skin immunology and CD1A-dependent T-cell responses, as well as Professor Suzanne Gabrielson, who is one of the pioneers into the research of exosomes in the immune response. And all those scientists not only helped us interpreting the data, but also provided us with some essential equipment at Cell so that we could carry out our experiments. And thank you for your attention.
The first question is there.
Hello, very interesting findings. So I have a question. Can you precise what is the molecular intracellular function of filaggrin, and can this explain why the fatty acids are shorter without filaggrin? So how can this happen?
Yeah, this is something we're really trying to find out, and we don't really know. We just know that the protein is really multifunctional, and there are many studies both in vitro and in vivo that showing this. However, we still have to kind of look into the exact mechanism of what's happening. But we really know that, for example, in filaggrin insufficiency, like the fatty acids of the lipids on the biological membranes are shorter, so this really affects the integrity of the epidermis. But you really need to look into the potential mechanisms.
Thank you.
Thank you for a very interesting talk. Firstly, I would like to appreciate that you did use MySEF terminology in terms of EVs, not exosomes, but small EVs and rich fraction.
Yes.
The second, my second remarks and the question is, you observed the changes in the lipid component in filaggrin insufficiency, and did you observe any changes in the uptake of these EVs to the cell, to the target cells?
Yes, this is something that we have as a kind of related but a separate project. And yes, we did. And our results, we showed that the uptake is actually decreased upon filaggrin knockdown.
Decreased. So thank you.
Hello. I have a practical question. So by using cosmetics, creams or ointments, are we sensitizing ourselves to the lipids present there or desensitizing our immune system?
Definitely sensitizing in many cases. And the cells that kind of produce the response are already there. So there is a paper Well, there's a few papers, but there's one from 2020, I believe, that identified multiple components of like cosmetics or personal hygiene products as being actually antigens for the CD1A. And they showed that they really do produce T-cell responses. So yes, cosmetics definitely make a difference, and they are active immunologically via the CD1A.
Do you have any idea, are the cosmetics screened for the shorter lipids or the lipids that could be especially potent at sensitizing immune response?
Your question is why some lipids kind of can facilitate the response and one and the others block? Is that what you're saying?
Well, that I assume that there is a difference, but
Yeah.
can these harmful lipids be somehow screened before they reach consumers?
Yes,
or
I mean,
us?
that would be the goal, to actually screen it. However, we're not at this stage because, as I said, there's not really many lipids that are known to be clearly permissive and non-permissive, as I said. So for now, we have an idea. For example, if you have lipids that have bulky heads, they can kind of stick out of the CD1A binding groove. And so the length of the lipid can make a difference. So this is something that we kind of know. But the tricky part also now is how the number of the unsaturated bonds makes a difference, because we see that in some cases the unsaturation actually binds them better and a different configuration of the CD1A. So it really varies, and we really need to test more ligands, because as I said, we even had some troubles to analyze our data in the context of what's already published. So definitely we need to test more ligands, and then we can draw some conclusions on which are more permissive and which are non-permissive.
I have one question.
Yeah.
Can I ask you if these results would be possible without international collaboration, if you will be allowed only to the national collaboration with some kind of embargo of contacts outside?
Well, you know, this is a very tricky question, and as a PhD student, I'm really lucky to be kind of on the science side, which is the fun part. So I'm sure the head of our lab can maybe, you know, explain better how it would be.
Thank you. Yes, I think we would really stumble, because even for the model that we're using, the cells, K562 cells expressing CD1A, which is essential for this part of the project, it wouldn't be possible to just, you know, in the project length to generate that. So we got these cells. They are generated in Harvard by Professor Branch Moody, but we got them via the collaboration with Professor Og. So this is a simple thing like that. We had a lot of trouble because with the lipid ligands generally and responses of T cells to that, to those ligands, they are not easy. We know very little about them in comparison to peptide antigens. And so even the methodologies are not there yet, like 100% and tetramer staining. So we were trying to do some of that. You know, this is the, again, collaboration with Professor Og, especially to kind of make this work even. But at the very, the example of the results that you could see there, the ELISpot results, which is the dotted wells where you could see cells reacting, we didn't have access to equipment for a short time. So we actually would make the plates and take them to Oxford, either, you know, personal routes or via conference meetings, you know, all of that. And that was read. Now we have the machine, so that's much easier. But yeah, yeah, so it would not be possible to detect those responses.
Okay, thank you. So let's thank again our speaker.
Thank you.
And now I want to invite Marcin Kuźniak, who made his PhD at Jagiellonia University and Paul Scherrer Institute. Then he made his postdocs in Canada at Queen's University and Carleton University, focusing on search for dark matter. After habilitation, he was promoted to associate professor at Nicolaus Copernicus Astronomy Center, and now he's leading a research group at AstroCert, and his group is collaborating within the Global Argor Dark Matter Collaboration.
Hello, everyone. I don't think these are my slides. So meanwhile, thank you very much for the invitation to give this talk. I'm here to talk about a somewhat different topic, astroparticle physics. I'm one of the group leaders at AstroCent, so let me introduce the center first. AstroCent is one of the international research agenda programs of FNP, led by Professor Leszek Roszkowski, who is also a leader of the theory group. We have also two groups which work on experimental astroparticle physics, liquid argon detectors primarily, and dark matter search, a group which works on gravitational wave detection as well as data acquisition and computing groups. We mostly study invisible universe, so hidden sectors of universe using neutrinos, gravitational waves, or dark matter searches, and we develop sensitive devices to do these searches. Our strategic partner is the French APC from Paris. So, well, this is kind of the questions that we think about. What is the universe made of? And many of you probably know that what we know from particle physics is in a drastic contradiction with the picture of the universe that we know from astronomy, from astronomical observations. First of all, we have no idea why there is more matter than antimatter in the universe. From particle physics, there is no reason for that to be so. And for the matter itself, for what we see in the universe, turns out that every particle, every component of matter that the science knows so far is only a small fraction of the whole. There is a so-called mysterious dark matter, which neither absorbs or emits light, that we know about from astronomical observations. For instance, observing how large structures in the universe look like. There is no way to explain that without the presence of something like dark matter. We know exactly how much of it there is. We know where it is distributed in space. We don't know what it is. However, people speculate that it is some sort of a new unknown particle, and that could also resolve a few other puzzles in particle physics. So how do we search for these particles abundantly present in the universe? Well, we go deep underground to special laboratories and run our detectors there. Why so? Interactions of these hypothetical particles with ordinary matter, which is what we look for, are extremely rare. If they were not, we would have seen them before in our other detectors on surface. So going underground shields these detectors from the main source of background, which is the cosmic radiation. 2 kilometers of rock, like here in a Canadian snow lab, which is a lab working in an active nickel mine, 2 kilometers underground is enough to get rid of that problem. So some of these labs are fairly large. The Snow Lab is one of the biggest one. This is where the Deep 3600 detector is working, a project that I've been part of since 2009, already as a postdoc. And currently our AstroCent group participates in data analysis analysis of data from that detector. The collaboration includes about 100 researchers from primarily Canada, Italy, but also Germany, Mexico, Poland, Spain, US, and UK. So DEEP is taking data. Another larger, 10 times larger detector of the next generation is currently under construction. It's a DarkSec 20K. It is built in a Laboratorio Nazionale del Gran Sasso in Italy. It's a highway tunnel under the Alps. It's a larger collaboration, global. It includes DEEP and several other groups which used to work on independent liquid argon programs. So 300 physicists, 60 institutions, 15 countries. Our primary partners, ours meaning AstroSense, are groups from INFN, GSSI, and APC, our strategic partner. Now, I need to introduce some technical jargon. these detectors are based on liquid argon. Okay, so we have a large volume of liquid argon. It is instrumented with some sort of photosensors. In our case, we use silicon photomultipliers, SIPMs. We have to use so-called wavelength shifting materials, reflectors or coatings. So this WLS abbreviation will show up. In case of DarkSide 20K, we are talking about 360 square meters of such wavelength shifting surface that needs to be made. So this is a big challenge. Now, why is it a challenge? Let me explain the measurement principle briefly. So, okay, now it's working. So inside of our liquid argon volume, which is the heart of the detector, we look for events like that. A dark matter particle elastically scattering on an argon nucleus and flying away. This nucleus acquires some kinetic energy, which is then deposited in the medium. And the property of liquid argon itself is that such energy deposits are then converted into scintillation light. Okay, so we look for tiny flashes of light coming from this liquid argon volume, which we detect with sensitive photosensors. Now, the issue is liquid argon emits very deep in UV light, 128 nanometers. There are no sensors which are sufficiently sensitive there, and this wavelength is absorbed in pretty much all materials. So we have to convert that to visible light. This is what we use wavelength shifting coatings for. So sensitivity of these detectors depends critically on mass of these detectors. We want them as large as possible, and on the light collection and detection efficiency. So our specialty at Astrocent are exactly these two things, so collection and detection of light. We develop novel approaches for conversion of UV light to visible light. The solution that we proposed and developed at Astrocent was adopted by DarkSide and is used in DEEP. There are hopefully some applications in photovoltaics. We'll explore that in the future. And we also work on SIPMs. We'll test a large fraction of modules for DarkSide in a dedicated facility in Warsaw at Cesamat, which is under construction. We do a lot of analysis and simulation. I will not talk about that today. So a few words about our key projects. So these wavelength shifter coatings, traditionally they are deposited with vacuum evaporation. This requires large vacuum chambers, is very time and energy consuming. We developed a traditional alternative approach based on using a large format foil, polymeric foil, which just has the right property. And we are working on enhancing this conversion efficiency even further, although even what's available on market after some adjustments is suitable at least for parts of the detector. So these 360 square meters will be made from that foil which we identified and demonstrated in situ in a prototype liquid argon detector in collaboration with our Italian partners. We built the device to demonstrate that here at Astrocent in the house. Then it went to a liquid argon detector at LNGS where it was tested. For DEEP 3600, we developed special wavelength shifting coatings with long time constants. This is something which allows to discriminate backgrounds. There are regions in the detector where, well, if there are any events from radioactive impurities happening in, let's say, so-called neck of the detector, it may lead to background events. So you want them to look different than the events you expect from the signal. It can be done by introducing a long time constant in the wavelength shifter deposited in that area, and then you can use so-called pulse shape discrimination to get rid of these events. We proposed the coatings, did some prototyping, actual production and testing was done in Canada and Germany, but we did the key part of the characterization here at Astrocent. Let me skip that one. Now, about the international collaboration specifically, well, as you see, we work with these large projects abroad in laboratories, which are only very few in the world. We need support to travel and be present there and contribute and make high-impact contributions and have our students and postdocs gain experience on site. And that was really only possible with additional funding from a European twinning project, DarkWave. We got about 0.9 million euro budget in a consortium with INFN, GSSI, APS, and TUM. We are the coordinator. This supports all our research-based travel for all astros and not only dark matter related. So in summary, well, I didn't talk about that too much. I decided to show things that we really developed here in the house. There is no dark matter signal detected yet in any of these detectors. We are scaling them up. Well, the ultimate sensitivity that can be achieved is for a detector 100 times larger than deep. Dark side is only 10 times larger. There is much ongoing and much needed R&D that we are leaders on, at least on the waving shifter part. International collaboration is absolutely key to make progress here, and I invite you to check other posters from other astrosand groups. There's one specifically on applications of SIPEMS to PET, as well as seismic sensors and other interesting material. Thank you very much. This is a picture from a conference we organized 2 weeks ago, gathering about 80 participants from around the world, 15 countries, specifically about technologies for light detection in noble liquid, noble element-based detectors.
There's a question there.
Thank you, Professor. I have a question, but let me introduce myself first. I am from Sano Center of Computational Medicine, and we're actually very interested in mathematical models that help make a contribution from a medicinal point of view or a medical point of view. And by analogy, like the folks of quantum computing have introduced themselves to be leaders for future computing and machine learning and artificial intelligence algorithms. Did you and your team design some mathematical models that can actually also lend themselves nicely to applications in medical and biology? That's my question.
So this is specifically about quantum computing?
No, I'm asking about if you're in your domain and studying the dark matters, have you designed any mathematical models or anything that can lend itself to understand the biology and the chemistry and our medicine better?
Well, I think the first thing which comes to mind is liquid argon-based positron emission tomography and related algorithms and software. So there is a poster which talks partially about that. I invite you to reach out to Masayu Kiwada here, show yourself. We have also a quantum computing group at AstroSand, also represented today by postdoc, and well, they use and develop quantum computing algorithms for things like analysis of satellite images. I presume for medicine, medical imaging interpretation, and so on. One could also develop either machine learning or quantum algorithm in the future. This is early days. But other than these two activities, we have no real contribution at the moment at AstroSand.
You're making a huge contribution in our future and understanding our universe, but I'm also a little greedy because our mission making contributions to the people who are living today as well. And I believe what you've introduced might also have a lot of contribution from a medical point of view. That was my question, but thank you so much. Appreciate it.
Thank you.
Can I ask a question?
Yeah, can I?
Yeah, but this was first.
Invisible.
First one? Okay, thank you. Thank you very much for the talk. I'm totally not an expert in dark matter. I'm just a laser guy. But I have a question, and I appreciate a simple answer. We know that there are other techniques of searching for dark matter, like Michał Zawada is using optical atomic clocks. We know that in Krakow, the guys are using magnetometry, the global network. So this experiment looks pretty complicated compared to the other ones. So where's the difference? Which one is better? Which one will finally lead to the results?
Excellent question. So I said it's probably an unknown new type of particle. Now, it can be at least a dozen different particles with completely different properties. So we have to take chances here and in parallel develop detectors sensitive to particular classes of these particles. So experiments with magnetic clocks are sensitive to a different candidate particle than what I showed. Our experiments are sensitive to so-called weakly interactive massive particles, WIMPs. Some people say they are favored theoretically because they can solve more problems at the same time, but I think there is at least another candidate axion which is at least as well motivated, and we have some sensitivity to axions, but there are dedicated experiments which do a lot better there.
Okay, we have 1 minute left for two questions that were already asked. So first is there.
So I have a question about your wavelength shifting coatings. How sensitive and efficient they are? Would they work with single photons, and would they work at different wavelength range, for example, that would be applicable in biology or elsewhere?
Right, so these coatings, well, the traditional vacuum evaporated coatings have been used for many years now, and yes, they have nearly 100% conversion efficiency, and yes, we care about every single photon, so certainly. These polymeric foils I showed, at this point, from what we get from a supplier, kind of technical grade foil, we can get up to 50–60% conversion efficiency. We think we can make it better, and we are starting to work with chemists and material scientists to push in this direction for larger detectors. The wavelength dependence is there, so it varies. At vacuum ultraviolet, we have these values I quoted. In near UV, it can be a little less or more, really depends on the substance.
Organizers allow for two more questions. So first on the back and then on the front. Yeah.
Sorry, I have a question which is most probably absolutely trivial to you, but could you disambiguate the word weakly? Does it mean that they interact via the weak interaction, or that they simply interact weakly?
Well, when the term was introduced back in the 80s, it was really about weak interaction, but by now more and more sensitive detectors eliminated that possibility. So it's not weak interaction at this point. It's
so that means that you search also for another interaction?
Well, Higgs could be the mediator. It really depends on the mediator particle. So
yeah, no, Higgs is related to mass, yes, so that could be the reason, yes. Okay.
So the class of models which was really the best motivation for these searches were supersymmetric models. So in a way, yes, it's a search for new physics. Whatever dark matter particle we eventually find will be new physics, will completely change our understanding of particle physics.
Congratulations.
The last one question in the front.
I'm just wondering how easy or how difficult it is to translate the know-how on development of detection technologies from the fundamental research, like dark matter search, to medical applications.
So there are several groups around the world which use either liquid xenon or liquid argon-based prototypes at this point, again with positron emission tomography in mind. Again, I invite you to see the poster, but yeah, there's more of that. There's more of that activity happening around the world. Having said that, it all started probably in the 70s, and still there's no commercial device on the market. So we think SIPIMs are really the game changer. This is a new technology, so things can accelerate now.
Okay, so thank you very much. And now I want to invite the last but not least speaker, Dr. Radosław Łapkiewicz, who made his a PhD degree under supervision of Anton Zellinger, this one that was awarded Nobel Prize this week at University of Vienna. And after that, he stayed some years at the postdoc stay in this university. And then he moved to the University of Warsaw, where he hits the quantum imaging lab at the Faculty of Physics. And for his work, he was awarded fellowship by the Polish Ministry of Science and Higher Education and Foundation for Polish Science.
Thank you very much, Michał, for the introduction. Good morning, everyone. So I would like to talk about applications of our research in biology, among other things. These are neural cells transfected with fluorescent proteins, and they are alive while being imaged. And biologists can somehow simulate the process of learning or of neural activity. And what is important here is that we see dendritic spines growth in this process. So this is what I hope will occur also in our heads during this talk, that we'll memorize something and dendritic spines will change. And also that many new connections will emerge during this conference, and the existing connections will strengthen. What is important here is the scale bar. It's 5 micrometers. If you remember, in optics, the wavelength of green light is about 500 nanometers. This gives us the resolution limit in the order of 200 nanometers. So dendritic spines are kind of on the border of what we can see with standard microscopes. And what we work on is we come up with techniques, or we use existing techniques to improve resolution of microscopy. And we want our techniques to be simple. So in this case, this was just recording a movie and analyzing the variance instead of taking the mean intensity, as you typically do, and you improve resolution and decrease the background as you see here. So this will be the subject of my talk. So going from blurry images to slightly sharper ones, using the tools of quantum optics, which is my background. So I'm the guy counting photons. That's why we needed strong collaborators on the microscopy side. And I was really happy that Jaron Zilberberg from the Weizmann Institute of Science agreed to work with us in the first team project. He supported it from the very beginning. I'm grateful for FNP for financing this, because this really strengthened our collaboration with him and his group. I'd like to also mention Dan Oron that took over the collaboration, and Ron Tene, who was the lead author from the Weizmann side on our joint projects. The collaboration is indeed very, very tight. So one example is you see here a picture from my lab, and what stands there behind is a part of a femtosecond laser. It's a huge device costing like $200,000, and this is not property of University of Warsaw. This is property of Weizmann Institute. They were kind enough to ship this monster to our lab, and it's still there. It helps you if you have to use public money to buy a laser. You can wait 5 years, then maybe it arrives. This was much faster. Okay. Right, and even more importantly, Anat Daniel, who is a former PhD student of Jaron, joined my lab and works now with us as a postdoc. Okay, I would like to pay tribute to Jaron Zilberberg. I'm really privileged that I, even though shortly, but worked with him. He was a fantastic scientist and really a great human. Like, he really saw a person in you from the first day, which, this is not always the case with brilliant scientists. He was really focused on people, and it's a huge loss that he passed away 3 years ago. Okay, so why break the resolution limit, right? You have a blurry image, you make it sharper, so obviously you know, this is exciting, this is obvious, this is one reason. If obviousness is not a good proof, then authority might be another proof. Other people also find it important. Even though this limit was written in stone and derived in the 19th century, it was finally broken, began in the 90s. And okay, so before we dive into super resolution microscopy, I want to remind you some basics. So in fluorescence microscopy, on which I will focus, you have a sample, and in the sample you have emitters like molecules or quantum dots. You illuminate them with blue laser and detect green fluorescence. Unfortunately, what you see from a point emitter is not a point image, you see a blob, you see a Gaussian blob, okay? And this limits your resolution. If two emitters are very close together, two blobs will start overlapping. This was derived by Abbe, And this d, the minimum resolvable distance, is roughly half the wavelength of light, 200 nanometers. Okay, so how to break Abbe's diffraction limit, right? He was a brilliant scientist, so there was no mistake in his reasoning. To break his limit, you need to break at least one of the assumptions he used. And this is the path taken by Hell, Betzig, Gustafsson, the authors of these famous Nobel Prize-winning techniques. But there was an implicit assumption also, this was 19th century, so Abbe considered classical light. How about, let's look what quantum mechanics offers. So if photons came in pairs and you counted pairs instead of single photons, the point spread function would shrink by a factor of square root 2. And this is quite a simple idea. A simulation shows that indeed you improve the resolution slightly. And this idea is due to Stefan Hell. And Stefan Hell is not famous because he plays saxophone, and he's not famous because he authored this paper, because such emitters don't exist. They are not practical. So this was a fancy idea, but didn't get him a Nobel Prize. Okay, what was important and missing in this idea is that you can equally well use sources of missing photon pairs, not photon pairs. And it turns out that missing photon pairs, that these sources are very common. Every molecule, every quantum dot, every single photon emitter is a source of missing photon pairs. Namely, it cannot emit two photons at the same time. This is bringing us back to one of the, or I would say the founder of quantum optics, and the guy who unfortunately didn't get Nobel Prize for his discovery. He died too early. He was the first to observe anti-bunching, the effect that a single photon emitter doesn't emit two photons at the same time. And this experiment nowadays became very simple. This was a pioneering experiment back then, but now it's simple and very common. If you do fluorescence microscopy, you have these emitters in your lab, and you do this even though you maybe don't care about this. To prove that single photons are emitted, you need to split your beam of photons at the beam splitter, put two detectors, and correlate the counts. Okay, and then you analyze your data. You see that two detectors never click simultaneously. You could multiply the columns in this table, and that's it. You then shift the delay, and you see how many pairs you have for a delay. What you observe is a characteristic dip at zero delay, so no two photons at the same time. And this phenomenon is more common than you have thought. I have here a physical system that everyone knows, but I'm wondering if someone has an idea, looking at the time scale of anti-bunching, what the system is. 24 seconds, characteristic time. So the system is basketball. So as you know, there is one ball, it can only end up in one basket. This corresponds to like two detectors in the photon case. Anti-bunching is just saying that photons behave like particles in this experiment. This is real data analyzed by Ron Tenne from NBA. Okay, so we took this anti-bunching phenomenon and the idea of Ossip Schwarz that this is equally good as two photon emitters, combined it with Stefan Hell's idea, and put it into a fluorescence microscope. Okay, and indeed, building such a microscope in which we have to replace the PMT that you're used to by an array of detector allowed us to break the diffraction limit. Maybe a short break, just to keep your attention to the end of my talk. I would like to. This is a photomultiplier tube that we replace with an array of detector, and this allows you to improve the resolution. Look at this beautiful Soviet technology. I hope it's as useless as other Soviet technologies. Nowadays it's important for us, I guess. Okay, so the take-home message here is you don't need to do anything fancy. Your biological sample labeled with quantum dots or molecules even does emit quantum light, and then you can use this quantumness to improve resolution. Okay, so this is one source of super resolution contrast. Another source is blinking of emitters. So whereas this quantum correlation leads to a negative, quantumness of light leads to this negative correlation, blinking, so turning on and off of molecules leads to a positive contrast. This can also be used for super resolution. And this is what we have shown here. And these are the people who made this happen. I'm running out of time, so I will speed up. The point is here that we use the standard fluorescent molecules, and you can improve the resolution. On the left, you see image scanning microscopy, which is already super resolution method, and then we improve it even further by using this fluctuation-based method. And this was made possible by the use of this device, developed in the Eduardo Charbon group. And I want to play a magic trick here. So the box is empty and the SPAD array appeared in your pockets, and this applies to the people who have iPhones newer than 2 years. So actually iPhones that were produced 2 years or later include SPAD arrays in themselves. So when we started, this device looked ugly. It's here, it had cables in every direction, breaking USB and so on. I believe that now this will be more and more accessible because of this boost from consumer applications. Okay. Then another important collaboration, apart from Weizmann, was with Professor Leszek Kaczmarek, Nęcki Institute, and with Ania Berun. I just want to quickly mention that we built a 2-photon microscope that was transported these 200 meters from physics to Nęcki. And we obtained first images. This is Paweł and Łukasz working. You maybe not find this exciting. For me, this is exciting, and for them as well. What you may find exciting is doing, quantum entanglement experiments on Canary Islands. But if you make a retreat, make sure to have some free time on the beach. If you don't want to make your PhD students look like this guy there in the middle. Okay, so last thing I want to mention is that I'm really happy that my supervisor got a Nobel Prize. And by the way, in this photo behind him, you see my first experiment from the PhD. Described here. And within the first team project, we continue working with him. And these are the 3 publications and preprint that we made as a part of this project. And this was again based on the idea of this pioneer of quantum optics, Leonard Mandel. Okay, and the most important slide at the end, I would like to thank my great team. So Monika Pawłowska, Nat Daniel, Adrian Makowski, Paweł Szczypkowski, Oleg Krupiński-Ptaszek, Bonik Shika and Sanjukta are actually here presenting a poster. And yeah, Bernard is a master student. Thank you very much for your attention.
Since nobody is yet brave enough to ask a question, I will ask first question. Do you think that this formalism that you use for this anti-correlation detection can be applied to other particles than photons? And I'm not even talking about, standard electrons, stuff like that, but maybe, some other kind of energy of waves of interaction, like, gravity or dark matter or something like that.
I didn't catch the beginning of your question. Which method?
All your formalism here is based on photons.
Yes.
Onto photons. Can be somehow used to other type of particles.
Mm-hmm. So I believe the phenomenon of anti-bunching would happen for any single particle. If it can be used in a given system, I don't know.
Do you need to, does the image, this quantum, let's say quantum image, need to be acquired like longer than the classical one?
Yes, that's the main point I skipped. This is the main weakness, and yes, so the price we pay is that we have to replace the detector and perform a longer measurement. This is this movie that you need to record and then analyze the statistics.
Thank you. So I had a chance at some point to be working on STED with a group of Professor Christian Egling, who was a student of Stefan Hell. Mm-hmm. And so the papers on STED, they say 8 nanometer sort of resolution. We could see up to maybe 40. So what is your limit you have in your system? And also whether with the STED, for example, the bleaching was a massive problem because of the powerful STED laser, bleaching this all out. Do you have the same issue in your system?
So I think it's not as severe as with STED. We don't need high intensities, but we do need to make the measurement for a longer time. So at some point bleaching becomes a problem. And on the first issue, we definitely cannot compete with STED, even on this 40 nanometers. We expect something like a factor of 2.5 compared to the fraction limit. So you end up at 100 nanometers if you're optimistic.
Okay, I see no more questions. So let's thank Radek once again. And before concluding the session, I want also to thank the members of the selection committee for this panel. Professor Agnieszka Błachnio-Zabielska, Professor Karol Grela, Dr. Tomasz Wróbel, Dr. Jacek Plewka, Dr. Marcin Śmiglak, and your humble servant here. So thank you very much, and thank you, our speakers, again.
Thank you very much for presenting your great research. Now, two important remarks. First one is that the abstract book is available only online. So please go to our webpage or to the application. There you can find the abstract book, and the abstracts online are also available. The other remark is I'm very happy that some of you have been rushing here to hear what we are doing and saying here, but please do not forget to go to the reception, take your ID and sign the attendance list. Now it's time for the first open poster session. I would like the authors of the posters to head to their posters, numbered from 1 to 40, and all the other participants to choose where to go, who to meet, with whom to connect. We will see each other in an hour. It is at 1:25 p.m. Enjoy your talks. Thank you. I am very glad to see you here again. I'll be even more glad if you would please take your seats. It's mainly because of our YouTube viewers, because we are streaming, and that's why we need to be quite punctual. So please take your seat and we'll start in a minute. Consequences are everywhere. Actions have consequences, and being able to learn from them revolutionized life on Earth. While it's easy enough to see that consequences are important, only a few heard there is a science of consequences, gathering fundamental learning principles that affect us every day with applications everywhere. Will the science of consequences apply to our upcoming session? Dr. Bartosz Wawrzynów, my colleague from the FNP, Tech Transfer Coordinator, is going to moderate the coming presentation session entitled Team Up Synergies Towards Science with Consequences. Bartek, the stage is yours.
Thank you very much, Kasia. I hope the microphone is operational. Ladies and gentlemen, it is my absolute pleasure to host this session. And what I'm really excited about is that we're continuing the trend in part from the previous session. I don't know if many of you have noticed, but an effective scientist needs to be a really good networker. And an effective scientist is a problem solver. And at the same time, an effective scientist is also a very good storyteller. And I truly hope that we in STORE have 4 stories that will be told by 4 spectacular scientists. So without further ado, it is my pleasure to welcome Professor Paweł Moskal. He's an inventor of positron emission tomography based on plastic scintillators and a method for positronium imaging. He conceived and headed a medical experiment demonstrating the first positronium image of the human brain. Naturally, he's a professor of physics and the head of the cluster of nuclear physics department at the Jagiellonian Universities. Professor Moskal is a laureate of the Foundation for Polish Science in the START, MPD, and TEAM programs. He co-authored 42 patents within the EU and the US and Japan, and has over 400 scientific articles. Currently, he's heading the JPET collaboration developing positronium imaging and constructing the cost-effective total body PET system. Professor, please, the stage is yours.
Thank you very much for such a kind introduction. I will try to show you what we are doing. And I will talk about first in vivo positronium imaging, which we managed to obtain recently, and plastic scintillators for tomography. So there will be like 2 out of many subjects we are researching. I have chosen 2 that you please remember. We are working on cost-effective positron emission tomography and positronium imaging. The second one is to improve the specificity of the imaging. Sorry, it was too quick. And since it could be that I will not manage to come to the end, I will start from the end. So this is the tomography system which we invented. You see a patient inside. So we managed to make the first images. So you see that the dotted lines are the photons going out from the head. The red one is the photon also going out from the patient head from the nuclear decay. Can you use the microphone? Okay, thank you. So this is anyhow not working. So the patient was administered prostate-specific membrane antigen, which has high affinity to prostate membrane cells. But at the same time, it happens that it also has a high affinity to the receptors at the cells of glioblastoma. And we used that fact. We labeled that with gallium, emitting positrons. And here you see a molecule, as an example, from that glioblastoma receptor, with positronium atoms, the atoms from electron and positron, which can be formed inside that molecule. So this is like intramolecular biomarker or intramolecular imaging what we developed. That positronium may decay inside these molecules, and we have invented a method which enabled us to measure the properties of that in a living organism. And here is an image, the lower one. The upper one is the standard PET image of that glioblastoma in the patient, which is also our first image with the plastic scintillator. And the lower one still blurred. This is the first of that kind which was obtained. And now I will explain it in more details. The JPET is from the Jagiellonian PET. Jagiellonian is the name of our university, but also the name of the king's family who sponsored the university. This is our best student so far. These are our students who will be famous, but we still don't know now yet what for. We are waiting for the results. The group consists of two departments. One is on around me, on the nuclear and particle physics, and the other part is from biomolecular and medical physics. We combine these two, headed by Professor Stępień. Here you see 5 postdocs supported by the team project and 7 PhD students. So we are very grateful for that support. Somehow not changing. The positronium, I will start from explaining the basics. Positronium tomography works by administering to the patient molecules labeled with beta plus isotopes. They are annihilating inside the body, as you may see at this animation. This is positron emitted from the nucleus and annihilate inside the body, mostly into 2 photons. We study also entanglement of that photons, which is something totally new because these photons are 1,000,000 times more energetic than the optical photons, so we developed a new method. This is not the subject of today's talk. But measuring 2 of them, one may plot a crossing of the lines, millions of lines, and get an image. This is an image of my head. So this is proving that I have a brain, and even more, this is that the brain is functioning. This is metabolic activity of the brain. Okay, to be more specific, as a scientist, it was in 2002 when I was a postdoc, so at least at those days it was working. Here you see how it works now. All that detectors are crystals. Crystals which are about 1/5 price of the gold. So the typical tomograph is about 2.5 million euro, and for the total body it could be like 12 million euro. So we were working on make it cost effective. Here you see our last solution. Most of the countries are wide at this transparency, which you see here, showing that this is not, most of the community in the world has no access to such a tomography. So this is the aim to provide the cost-effective tomography. We use plastic scintillators, which are about 50 times cheaper than crystals, making the tomography as a whole about 5 to 10 times cheaper. The crystals cannot transfer the light that good as plastics, so use these scintillators also as the light guides. But this should have not worked. So in the textbook, you find it will not work with the plastic scintillators. So we need to develop a lot of methods. One of them was developed by the postdocs here, Sushil Sharma. I will not go into the details, but I can, if you would ask. We developed like about 40 methods to come to the end in the patents, which resulted also in quite recent publications. And this is like a long adventure. So starting from 2009, we first proof of concept, which were a kind of misusing grants from Meson production. Then we came to the first prototype, which you see here. And I will show results from that prototype. This is what we have in the lab. These black strips are plastic scintillators. This is, again, a postdoc of the team who was most involved in that studies. This is how it looked like in the real life. So our first image was the person. This is how physicists see a person, the cylinder. Annihilations were at the surface, and we proved that it works. We made image of the surface. So now positronium. If one look more closer to what happens in the body, so please look more closer, you will see that this annihilation is not immediate. It takes some time. So it takes time because in about 30 to 40% cases, there is an atom formed inside the body. And this atom may live in the body. Its lifetime depends on the intramolecular structure. So here you see an example when it may interact with oxygen, it may interact with the surrounding molecules. So the lifetime will depend on the structure. And this is what has also consequences for the pathology of the tissue, what we are studying. Here is an example that measuring the lifetime, we may look for the differences in the pressure of the oxygen between healthy and cancerous tissue, just as an example. One of the things one may measure is the ratio between the decays into 2 photons to 3 photons. And again, here we have made a first image of the multiphoton tomography. This was the first one. We have also shown that we can measure with the detector the lifetime of these atoms inside the body. So first here inside the cylinder. Here you see a typical lifetime spectrum with positronium atoms short and long-lived. And then we came to the imaging of ex vivo imaging of the tissues. Here you see 2 tissues from the cardiac myxoma operated from the patients and adipose tissue. SALT was used as pharmaceutical, and this is an image here. We obtained a normal image, and for each voxel of that image, we could measure the time between the emission of the photon from the nucleus and the annihilation photons. And for each voxel, we could measure the positronium lifetime. And this is the image of that positronium lifetime, which you see here below, the first of that kind. We measured that for many patients under the guidance of Professor Stempins, and for each patient, it was a huge difference with observable difference. Then we went to the in vivo imaging, and this is the most important now, so I will skip. For that in vivo, we need a detector which we can go to the hospital. First, we went to the tomography center. This is the reintegration, joining the reintegration program with Antoni Ruciński. In the tomography center, we've proven that with the detector, we can monitor the beam range by the irradiated patients. You see it here. And then we went to the hospital. And here you see the most important from the hospital. I will skip some details. So again, positronium is produced inside the body. We use gallium, and we built that detector at the hospital. And here you see, we had 10 minutes to make an images from the allowed by the ethical committee. And here you see how it works. The tomograph behind is a Siemens, 2 tons. This is our 50 kilograms. We could move it, change the shapes. And these are this is now another action with the patients you have seen at the beginning. So this is now what I explained at the beginning, that we could measure with the tomograph the standard images, which you see here. This is a comparison between Siemens and ours. We have now 10 times less statistics, but anyhow, everything is like above is Siemens, below our figures. And this is then the image, the one below, of the positronium inside the human body. It's low statistics because of two things. One is that our present system has 30 times lower sensitivity that it can, 30 times, and the other, that gallium has only 1% of that decays which are useful. So this imaging in the regular tomography, having a total body PET, which we are now working on, could be done with the 3,000 times more statistics, 3,000 times. So this would be then regular images, and now we are working on that total body PET with postdocs and PhD students from team, and these are designs, some work from the lab, and this is what we are now constructing. Completely new design. The patient will be lying on the bed, not moving. This is a standard CT, and here you see our tomography system, which will cover the patient like with solarium. This is light, many times lighter than the standard PET. And if we manage to get funding from the ministry, which we are now fighting for, this would be the first PET, not only in Poland, such kind, but also in Europe. Thank you very much.
Thank you very much, Professor Moskow. And thank you to the strict regime. It's very difficult to tell a story in 10 minutes. The room is open for questions, if there are any questions from the audience.
Hello. Thank you for a nice talk. I wonder, the plastic scintillator is kind of lighter than crystals. So I think the energy resolution must be worse than the crystal. And my question is, how crucial this energy resolution for your system?
So thank you for that question. So it's even worse. There is no energy resolution, there is no photoelectric effect, so therefore there's 40 patents. So we need to devise the methods which are completely different from what is used now. We are based mostly on the timing and the position. The position axially we define from the times, difference on the two sides, and the position vertically is defined from the size of the plastics. So the position we have with the same resolution as standard PET systems. We have no photoelectric effect, so we have to devise the method for selecting scattered and not scattered photons based on Compton spectra. So we don't use photoelectric effects as are used for the high resolution, or maybe not high, but good resolution PET tomography. We are using continuous spectra with no need for the energy resolution. So we make a windows on the energy on that spectra and timing. And the efficiency, we can increase the efficiency with respect, because they are lighter, as you told. So the density is 7 times lower, which means 7 times less electrons per cubic centimeter. For 2 photons is like 40 times less sensitivity. But we could use more layers because most of the photons are going through. So with the geometrical axial enlargement and with multi-layer system, we can come to the sensitivities which are even higher than the current PET systems. 20 centimeter long. If we would have that PET, which I have shown you on the last figure, the sensitivity would be 20 times higher, which would allow to make movies of the pharmaceutical in the body.
Okay, thank you. Professor Wojtkowski.
Okay, thank you very much for nice talk and very inspiring work. I was wondering, because now the critical thing is to combine MRI and PET. So how your system, is it possible to have a compatible systems, or is problematic?
Yes, so this is the only system now in the world which is at the same time an MRI insert. So maybe you have seen that the animation, that this is our plastic scintillators, 50 centimeter longs, with the electronics which is based on the FPGA field programmable gate arrays, and silicon photomultipliers not so sensitive to the magnetic field. And plastics are like 2 centimeters of fat in the human. So we can insert that in the MRI and make simultaneous images. And we took care that there is no any metals in that design.
Thank you. Thank you very much. That's all the time for the questions that we have. So, Professor Moskal, thank you very much.
Thank you very
I understand
much.
you will be available for questions in the background. And now I would like to introduce Dr. Anna Kajetanowicz to tell another story. She did 2 postdoctoral fellowships, first in Karol Grela's group in Warsaw, and the second one with Thomas R. Ward in Basel. As she returned to the international, the ICHO, Polish Academy of Science Institute in 2015, she transferred to the University of Warsaw, and at present she's the deputy director of the Laboratory of Organometallic Synthesis at the Biological and Chemical Research Center. Dr. Kajetanowicz, the floor is yours.
Okay, so but I think that we have to switch the presentations. Okay.
Okay, apologies.
Okay, so thank you very much for a very kind introduction. And I am working on the TeamTech project, and together with the head of the grant, Professor Karol Grela, we are searching for catalysis for the 21st century chemical industry. And in short, olefin metathesis is a very useful reaction which allows for a reorganization of substituents around double bond. And depending on the substrates used, we can obtain a plethora of different compounds, including cyclic compounds, polymers, microcycles, and also linear olefins. And the value of this methodology was awarded with Nobel Prize in 2005 to 3 chemists, Yves Chavin, Robert Grabs, and Richard Schrock. And although this methodology allows for creation of really very complicated molecules, as the reaction conditions are typically very mild, and the currently used catalyst tolerates lots of functional groups, but it's still not widely used in chemical industry. There are some precedents, but in general, so those catalysts are not very well recognized in this society. And there are lots of problems connecting with the more general use of those methodology. However, I think that the most important are the necessity of usage of aromatic or fluorinated solvents, the low stability of a currently popular catalyst in the presence of air, moisture, and also ethylene, which quite often is one of the main byproducts of the metathesis reaction. And this causes low activity of the catalyst, low selectivity, and required high catalytic loading. And also, especially what is especially important for pharmaceutical industry, so especially when the high catalyst loading is used, so the traces of ruthenium stayed in the final product, which of course is not possible to use then in active pharmaceutical ingredients. So within the project, together with our scientific and also industrial partner, we are trying to solve some of those problems. And the first story will be about the metathesis in the presence of complexes based on unsymmetrical and heterocyclic carbene. And this is mostly cooperation with Apeiron Synthesis and Polpharma company. And those type of those type of complexes has 2 different I think that it will be easier to do it here. So those 2 catalysts have different substituents on 2 different nitrogen atoms in imidazolinium ring, and this causes much higher stability of those type of complexes in comparison to their symmetrical counterparts. And we developed a large family of such complexes, but today I would like to concentrate on the one with thiophene moiety. And as from preliminary study, we knew that this catalyst exhibit very good properties. So in cooperation with Apeiron Synthesis, we obtained this catalyst on the large scale. So starting from simple and relatively cheap starting materials, after optimization with high yield, we were able to obtain like half a kilo of the catalyst. So you can imagine that when we are working in a small scale, so this was really high, a very huge amount of the material for the research. And to show you main differences between catalysts with symmetrical and heterocyclic carbines and unsymmetrical ones, so I would like to show you comparison between Hoveyda, so one Hoveyda type catalyst, one of the most popular complex and commercially available, and our system. And as an example, I would like to show you the reaction of octene, alpha olefin, which is especially, this type of complex of compounds are particularly prone for migration of double bond during the reaction. And as a result, in the reaction mixture, we observe not only the products, desired products of olefin metathesis reaction, but also products with shifted double bond and also longer and shorter homologues of this kind of complexes. And just to show you about the selectivity, so in case of our system, so the catalyst with thiophene moiety, we obtain selectivity on the level 98%, so almost full selectivity, while with Hoveyda second generation, half of the product was not, half of the products were not the ones which we expected. So during this project, we also introduced new solvent to olefin metathesis reaction, and this is for metal tetrahydropyran. So it's quite similar to THF. However, the advantage of using this solvent is that it's not mixed with water, and this really facilitates the purification of the system. And here, so using this solvent, we were able to perform the reaction of different classes of substrates, including easily isomerized substrates, possessing lipophilic, polar, or really fragile group. And also with this complex, there were not any problems with synthesizing a family of biologically active compounds. It's analogs and precursors, and using rather low catalyst loading, we were obtaining a huge amount of complexes. And also we were able to increase the scale of using this reaction up to 33 grams. And here I would like to point out that one of the most important things was that the amount of ruthenium in the final product was only 88 ppm, just after simple filtration of the product from the reaction mixture. So another story will be using catalyst immobilized on different supports. So we are using many of them, but here I would like to concentrate on cooperation with Professor Michał Chmielewski, and he is preparing metal organic framework for us. Introduction of the catalyst on such a support is very easy. So you just have to mix the solution of the catalyst with the sample of MOF, leave for a while, like 30 minutes, and you obtain the pure complex. Then such a complex can be obtained in olefin metathesis reaction. And here in the project, we started from checking what kind of solvent will be the best for such a process. And we compared both conversion and also the ruthenium content. And we were lucky, as one of the best results we obtained for dimethyl carbonate and also for acetone, as the conversion of the starting material was very high, and also the level of ruthenium in the final product was very low. And also here we were able to use this system for synthesis of many compounds with biological activity or with potential biological activity, and also using up to 0.5 mol% of the catalyst, we obtain a large family of that kind of compounds. Here also important was that in each case, actually, so almost in each case, we obtain a very low ruthenium content, which can be even lower when we used ruthenium scavenger. I will show you the structure on the next slide. And here I would like to concentrate in the last 1 minute. I would like to concentrate on metathesis in flow, and this is more recently is more and more popular reaction, as this process in general, in comparison to batch processes, is much cleaner, safer, and also is very easy to scale up as you just run the reaction longer. And you don't have to take into account different energetic and mixing processing occurring in a batch reactor. And this was in collaboration with Professor Vuji from Singapore, and we developed the system. Then again, we obtain a series of biologically active compounds, and here in almost all cases we use 0.3 mol% of ruthenium, and the residence time in the reactor was 5 minutes. So in a very short time we were able to obtain such system, and in all cases the amount of ruthenium in the final product was very low. Also here we performed the reaction on a large scale, just running the reaction for 12 hours, and we were able to obtain up to 62 grams of the final product, sildenafil derivative, but of course running the reaction longer will improve that amount significantly. So there are lots of problems related to application of olefin metathesis into chemical industry. We are trying to solve many of them, so concentrating either on special carrier or different more green solvents, or removing of the ruthenium from the final product. And I would like to thank people involved in the group, Foundation for financing this project, and you for your kind attention. I'm happy to take any questions here or during poster session.
Thank you very much. Are there any questions from the audience? Given that I don't see any, I can take this advantage and ask a very basic question. The question is regarding the partnerships with an industry like Polpharma, and the dialogue is with you as a basically R&D unit. Do you, is that dialogue open, or is it more of a, they have a wish list from 1 to 10 of what would they like to do?
No,
or what would like to have, and you provide that we can do this in a given particular amount of time. I'm very curious how this knowledge exchange framework within our local, at this side of the pond, in our neck of the woods, how does this work?
Actually, so the dialogue was open, and we first had a look what kind of processes they have in the factory inside. And then we checked in which ones we can take part, analyze them, and then try to obtain the molecules that are producing, they're interested, using olefin metathesis. And then we are also checking what is the cost, what is the price of the current process, and what could be the price of the process performed in the presence of olefin metathesis catalyst. So for now, the balance is not really good. However, I believe that with the development of better catalysts, and also, as I said, so part of our work was application and development of new green solvents. And this is for now especially important, as most aromatic or fluorinated solvents are harmful for the environment, and there are their usage is restricted or can be banned even in the future. So, like searching for good alternatives here can be really beneficial.
So, apart from classical chemical work, you're doing translational and championing work of presenting new technologies into a system which is not very open or very, well, it is compatible, but it's not hungry for the new wave of catalysts, and you're showing a new way. So bravo, and thank you very much.
Thank you.
Our next speaker, scientist, storyteller will be Dr. Marcin Pawłowski. Marcin Pawłowski obtained his PhD at the University of Gdańsk in 2010, and shortly afterwards he was awarded the EPSRC scholarship at the University of Bristol, where he worked as a postdoc. In 2013, he returned to his alma mater, which is a very good choice of a city, I must say. Currently, he's the head of Quantum Cybersecurity and Communications Group at ICTQT, a center of excellence solely financed by the Foundation of Polish Science IRA program. He is also one of the founders of Secure, a company that deals with quantum security and quantum cryptographic solutions. Apologies for my lag. Dr. Pawłowski, the stage is yours.
Thank you. Thank you for inviting me here. I would like to talk about this little beauty. It's SecureRNG. It's a quantum random number generator. It's a purely quantum device, meaning it uses quantum effects which cannot be modeled by classical physics, and as such is one of very few devices in the world. There are more or less a dozen of companies which build things like this, including Google or IBM. So we're in a really good company with our little thing, and we're obviously very proud of. And this device is a random number generator. It simply produces random numbers, which is extremely important. According to Polish law, every device which processes information with security clearance of classified or higher needs to have a hardware random number generator like this built in, because these random numbers are needed for many things, but the most important use is to safely generate cryptographic keys. And if you don't generate them safely, then it's very easy to hack your communication. And let me just show you something. So on the left-hand side here, I have a list I have taken from CryptoStack Exchange. These are results of using Euclid's algorithm. It's a 2,500 years old algorithm, which can be used to break public keys in such a way that you can get private ones. Don't try to read this. I can send you this. What I wanted you to is to impress you with how many attacks were conducted, and what is said there in very small letters is that usually around 1% of the keys in public repositories can be easily hacked, and only because bad randomness was used to generate them. The Euclid algorithms is very cool in a sense that we don't even know what was wrong with the random number generator, but since two keys were too similar, we could break them. We know that something was wrong. On the right-hand side, I have something totally different. This is a diagram of a dual elliptic curve pseudo-random number generator, which was standardized and introduced by American National Institute for Science and Technology. And later, due to Snowden, we found out that actually NSA designed it in such a way that they could easily hack it, and then they actually paid producers of cryptographic solutions to use this random number generator as a default and in their systems, so they could easily eavesdrop a lot of communications. And so how do we solve this problem? So we see that random number generators can just fail because they are bad, or they can be made bad on purpose. And quantum random number generators are a solution, but they come in two flavors. One is you take a random number generator and you just put a quantum sticker on it. And this is actually, unfortunately, legal. You can do this just as Finnish Quantum does. But if you do this, you can claim that you have also a quantum computer, because actually transistors in your laptops are using quantum effects. So it's really difficult to prove somebody that they shouldn't be allowed to call their device quantum. On the other hand, we have devices which I would say are really quantum, like the one on the right-hand side. And this is an immensely complex device. It was built and tested in 2010, and it uses traps with single ions which emit entangled photons, which are later entangled themselves in a procedure called entanglement swapping. If you heard the laudation for the Nobel Prize was for the entanglement swapping. So it's really complex physics. You don't need to understand it to know that it's complex. The device was the size of a room. It was very expensive, and it generated random numbers with a speed of 1 number per several minutes. So it was useless. So on the one hand side, we have really bad devices. On the other side, we have really complicated devices. So first team, funded by a Foundation for Polish Science, comes into play. And what we want to do is we want to do proper random number generators without compromising their security. And what you see below is actually a diagram from our patent application. This diagram has no relation with what we wrote in the project proposal. In the project proposal, we actually had 2 ideas for patent applications, and later we found out that this one is better than both of them. But in the project, we promised 2 patent applications. So again, I go and talk to Anton Zeilinger and ask him, what do you do in this case? And he says, Marcin, whenever I write a project report, I'm writing, although in the call we wrote that we'll do this thing, we actually did that thing, and you must understand that it was much better. So I called FNP and asked, can I do this? And they were really a little reluctant at first, because they said, but you promised 2 patents and now you want to have only 1. So I say, I'll sweeten the deal with 3 more publications. And then they say, yes, fine. So this worked. And then when we have this, we decided that this is a great idea, and we have founded a startup. So the patent was actually by Gustavo and me, but we couldn't start a company ourselves. So Steve there is our electronics guy. He's a brilliant professor and know how to build this. And Jose, although he has a PhD in physics, he's actually a businessman. It was not his first startup, so he knew how to do this. And then as Jerry Maguire said, show me the money. You cannot do anything without the money. And the rule of startups is go to investors as late as you can. Going to investors will cost you shares, and shares are your most valued commodity. Don't do this. So use as much public funding grants as possible, and we were able to get rooms and devices from which was worth more than $2 million from University of Concepcion, where Gustavo works, and we used some of his other research grants than FNP team. And then later we actually got an investor, but this investor decided that they, instead of shares, they want to get commission from our sales, which was also really nice. And this was enough to build the first prototype. This is not actually our prototype. We don't have any photos of it. It was dismantled, but this is its visualization. But worry not, based on this prototype, we built this. We built this pre-commercial version. It was tested by Loteria de Concepcion. It is the first lottery in the world which used our numbers to generate draws, and then this was dismantled, and now we have our version 2, the device which you can actually buy, and some people are buying. We have our first sales, so we're very happy of it. And then this is the future. So the first idea about the patent was in 2018. Now we have our products, and what we want to do is we want to minimize it and start using it, selling it on a wider scale, and so on. And now I have some further developments. So this is an expensive device, and if you want to play with it and you're not that worried about security, you don't really want this in your room, so you can use this. This is me on Tuesday, nice socks, building another thing. This thing you can see here is a quantum random number repeater which takes numbers via internet from our device, which is sitting somewhere in Chile right now, and then mixes it with its own random numbers in such a way that we are not losing security, but getting extra fluidity of these numbers. They are more stable. And then this is quite cheap device, and everybody can have it and plug it in your computers. And I don't have it here because it's actually now being demonstrated in InfoShare Expo in Gdańsk. And the second thing that we can do is, this is a website of Polish company Quantum Blockchains, and they are providing randomness from different sources of randomness on demand. And you can use ours, and here's a screenshot. You can see execution time on the bottom of almost 3 seconds. This is how much time it takes for the numbers to go under the Atlantic Ocean, through the Andes, Patagonia, and back. And as you can see, these numbers it generates are really random.
We need to wrap up. Thank you.
Thank you.
Ladies and well, thank you for that. We did not practice this. Ladies and gentlemen, we have time for one question from the audience. Nobody interested? Good. There is all.
Hey.
Marcin, could you explain the idea of your quantum number generator?
Okay, so basically it has an interferometer, and we could prove that if you have an interferometer, you can sometimes direct whole signal into one direction, sometimes in the other, and then you could also split signal half-half and generate randomness from where the signal actually happens to be, because there are single photons. And then we can prove that if, in the case where you expect photons divided half-half, if they are not really divided like this, then you would see also errors in something which we call testing trends. So when we expect all the light go in the one direction, we will see some photons in there. We have just mathematical formula combining this, and our device is just an interferometer in a box.
Thank you for that. Thank you. Thank you for the presentation. And ladies and gentlemen, we're slowly approaching the end of our session. Nevertheless, it gives me great pleasure to introduce Professor Maciej Wojtkowski, who's super active in the field of biomedical imaging. And I mean, it gives me, again, great pleasure. An author of more than 250 publications. He's currently the head of the Department of Physical Chemistry at the Biological Systems at the Institute of Physical Chemistry, Polish Academy of Sciences. Recently, he also started a new center of excellence of the International Center for Translational Eye Research. And I had a sneak peek because he was one of the few speakers that actually was willing to share his presentation prior to this meeting. I had a sneak peek, and you're in for a treat. So without further ado, the floor is yours.
Thank you. So ladies and gentlemen, we are lucky or unlucky to live in very interesting times. So scientific advances from 20th century was translated to really incredible acceleration of technology that we can experience in our everyday lives. And one of the most beautiful example of this is ophthalmology. As you may know, the first surgery in ophthalmology was performed 4,000 years ago in ancient Egypt. And then, since that time, till our times, it was not much of improvements. And currently, it is incredible because ophthalmologists can treat blindness, inherited blindness in kids. And these kids can see. They can grow tissue and implant it to the human eye. And this tissue is further growing and replacing existing, destroyed parts of eye and can function. So this is really amazing. But for all these advancements, there is a need to develop technology that will support introduction of these new therapies. Simply by validating, by quantifying the effects in human eye. And there is still a lot of missing instruments that can be built and developed. And this is the main task of our International Center for Translational Eye Research agenda that we are developing under umbrella of FNP and European funds. So I was supposed to give you our research in the context of technology development, and actually I have to start from the definition of TRL. So this is technology readiness level introduced by NASA in the 70s. There are a couple of points that just correspond to the certain advancement of our technology. There is a division into 3 sections: research, development and deployment. For more advanced technology that requires to build up the market, the phase of development can be split into validation and valorization. And in our small center of excellence, we are working on 12 projects. Some of them are still on hold, waiting for more financing. And I have no time really to go through all of them. But we are trying to fill this vertical integration mode, meaning that we really want to conduct studies from the early research up to the validation phase, let's say TRL7. And so I decided to show this the technology development in context of some assessment method that I copied from Sarai Kemp, a person who works in one of the Israeli incubators. And she claims that there are 4 elements that are really critical to assess whether the technology can be successful in further commercialization. So there's market and need, solution and technology, implementation, and freedom of operation. So I decided to put our technologies related to functional imaging. It's only 2 projects from what we do in this plot. So it's just an exercise. I did it for the first time, and it was a lot of fun. So when we look at the need and market, it is quite easy to define because in ophthalmology, there is this visual acuity test. So the thing is visible here. There is just a board with letters, and ophthalmologists are asking patients to which row is still visible. So it's extremely primitive test, but this is the only test, functional test, that is done in 100% of cases in the regular ophthalmologic practice. We have more advanced instrumentation like visual field examination, which is 10%, and electroretinography and others that are only done in 1% of cases. So this part is completely not significant. But if you imagine all these new technologies, the therapies that are coming to ophthalmology, there's really need to look how our eye is functioning, not how it's built. So for that, our goal is to change this situation. So to introduce new techniques that will be used in, I don't know, 90% of cases, and that will give ophthalmologists new information about how the eye can work, and also all the pharmaceutical companies and all the companies introducing new instrumentation for surgeries and so on to track, monitor the progress in their therapies. So when we go to solution technology, there are 4 points. So we must address the need. We have to find or indicate the uniqueness of our solution. We have to give a supporting evidence and give sustainable assets. So in our case, it's quite easy to address the need because what we do is just a conversion of existing technologies to new, somehow upgrading them, but using completely different approaches. So in case of this fluorescence imaging, we want to image human eye also with fluorescence, but now having access to active retinal pigments. So those compounds, chemical compounds that are actively taking part in the vision process, because this is, up to now, it's impossible. And replace electroretinography, which is very coarse, very hard to maintain system, into new optoretinography that will be useful, accessible to everybody, and can be done very quickly and cheap. So that's all will help all these novel therapies that are currently introducing. So with these new methods of functional imaging, we identified the mechanisms that we can use for that, and we demonstrated for the first time 2-photon retinal imaging with this group of people. And for that, we needed a laser that was built by our collaborators from Wrocław Technical University, and even we had a small contribution to the development of the company because we created a need. And another technology that we are developing, so the movie is shown here, we are illuminating human eye with short blinks of light, and then people realized this 6 years ago, that our photoreceptors, so the photosensitive cells in our retina, they are responding to these flashes with a very tiny change of their thickness. And this tiny change is only 1 single nanometer. So now you can imagine how challenging it is to measure 1 nanometer change in the living eye, which is not cemented to the system, which is moving and so on. So we developed a technology called spatiotemporal optical coherence tomography that enables us to get this high stability and high speed. And we improved contrast of imaging that we can beautifully see different layers of the retina and even go underneath the retina for the first time to separate different layers of choroid that is supplying with the nutrients our photoreceptors. But what is the most important is that we see a very high contrast in photoreceptor layer, and we can then determine this tiny change of its thickness, up to even less than 1 nanometer, as you see here. And we can apply different stimuli with different frequency and look how our retina responds for that. So we have the supporting evidence. We have more than 30 papers related to these both technologies. We have sustainable assets. I will not read this. But what is really important is the freedom of operation. So this is one of the critical aspects. And when you think about this and take into account all parameters, it is very hard to have a full freedom operation in public institutions and with public money. So also concerning the IP protection, we have a way of assessing what kind of protection is best, either covering the broad aspects of your invention with the different lifetimes. So for short lifetime, it doesn't make sense to have a broad coverage because it will not work. For narrow coverage and short time, it is quite questionable to use such patents. And of course, the strongest one will be the general patents that will cover the entire method. But it's terribly hard to protect them. And these kind of patents we can generate in our institutions, research institutions. But as I said, it's very hard to protect them. And the other assets are patents that are particularly about some very detailed solutions. And this can be done in companies. So the roadmap for our development is following. So we are now in this TRL 4, but we believe that this is the demarcating line that we should leave academia with this idea and go to find the support from non-public investment. So coming to the some limitations, I was asked also to mention about in my opinion, what are the limitations in commercialization. So in public funds, we have these points like overregulation in grant arrangements, inertia resulting from monstrous bureaucracy that I don't have to probably comment here, inefficient system of public procurement, and what is most important is this unrealistic and detrimental expectation of rapid enrichment of research institution without investment input. So the problem is that idea is cheap in our times. So if we just have idea and even protect it with patents, for companies, patents at academic centers are the best freezers, as you can imagine. So they will be not interested to pay for these patents because they know that academic institutions will not fight with the infringement or whatever. So that's why these expectations are, in my opinion, unrealistic. For non-public funds, there are also some limitations like unrealistic time expectations, limited long vision and social responsibility of investors, and also lack of direct involvement. So to help that, I just copied this from Israeli case. So Israel is very productive, and they converted their public incubators into privately owned incubators, and there is 18 government license with competitive awarded and so on. And this works pretty well. So when I ask people from Israel, from France about the situation, they're saying, oh, it was, in our case, it was the same in the 80s and 70s, so it must change. But the question is if it really must change in Poland. So maybe that requires a lot of fight of all of us, to be a little bit more resistant to bureaucracy, to this all sometimes not clever or not logical solutions that are proposed. Okay, and with that, I would like to thank my group, my collaborators, sponsors, and thank you for your attention.
Thank you very much. Ladies and gentlemen, we still have time for a question or comment to what Professor Wojtkowski just said. So,
congratulations. I would like to ask, how do you disentangle between the eye movement and this 1 nanometer receptor change?
Okay, thank you for this question. So simply, we are doing the measurement that is in reference to other layer, but still we need to have extremely high stability of phase. That's why we use a very high-speed camera that enables us to measure 60,000 frames per second, and in full field, so that each pixel register the phase, interferometric phase, stable to other pixels. So that gives us this possibility really to analyze, and then we do extra, because there is blinking, we do extra Fourier transformation that enables us to filter out noise.
Thank you very much. Is there anyone else? I mean, I have tons of questions, and I will definitely ask them once the microphones are off. But one which is more of a PG-13 question is, I'm searching. No, one that is, the modes of financing. When you're entering this stage where you see that you're at a certain, the proof of concept is there. You're building a business model, and you're weighting the cost of capital. We just previously heard that grants, yes, they are cheap, they're equity free. They have their limitations. You have to do 7 patents. If you don't do 7 patents, I'm sorry, that's it. And then you have equity financing. But the question is, have you found smart investments in Poland that actually understand what you're doing and bring something other to the table than, a big pile of cash?
Yes, we found.
Okay.
But I don't want to go deeply into detail.
PG-13.
But this is also, very coherent with what I had as a feedback from researchers from abroad, and also what Marcin was mentioning in previous talk, that there is this stage that must be based on some kind of friends' money, or our own money, or money that are not really VCCs. And this is the hardest stage in each startup. But there is this stage that you have to invest your time, your life, and your own money.
Commitment?
To progress. Yeah. And this is something what is unfortunate. And this was the same what Sarai Kemp from Israel was showing, that in this first stage, this is really necessary.
Fantastic. So, Professor Wojtkowski, thank you very much for your time, for your presentation, for your input. Ladies and gentlemen, I couldn't plan.
Just ask a question.
Oh, apologies. Yes.
So it was.
But the microphone helps. Yes. Yes.
Okay. Yeah. So Jose Souza from Sano. You mentioned Israel as an example of development around startups and how they built all the ecosystem. But I think that there is a fundamental difference there, is that they brought VCs, hospitals, and research together, what they call this very interesting hub research center. So I think that that's the way, but what you see in Europe is, first, in our case, that we are working on personal data, clinical data, is that the connectivity that you get on those environments is completely different in relation to the connectivity that you have when you have to develop your own network with the hospitals in Poland or anywhere else in Europe. What is your idea about that? Is that a fundamental thing that can be taken as an example?
Yes, thank you very much for this remark. I think yes, this is one of the elements, right? It strongly depends on the technology, but of course it is very helpful if it works like this. There are many different ways of doing this, of finding collaborators. In general, if you have a product, there is not much problem of having these contacts. So after this first phase, that is a lot of investment and a lot of pain, it is possible, but of course it would be easier if there is this infrastructure set. Then it would be just easier and quicker. So that's true. But it also depends on the technology. So.
Thank you. If we're done, I will thank once again Professor Wojtkowski. Ladies and gentlemen, once again, we're approaching, well, we're at the end of this session. I would really like cordially and openly to thank our speakers for adhering to the time and at the same time being open and sharing candor when it comes to their struggles in the scientific journey. On my end, what I can only say is that this panel seems to be perfectly in line with the upcoming, well, this session seems to be perfectly in line with the upcoming panel post-lunch, as we really want to dig deep into the intricacies of actually setting up your own entrepreneurial activity within the academic campus and seeing what are the painstaking, what are the real problems in that process. But till then, I wish you a very short queue to whatever food sustenance you're aiming for. And I will be signing off. Thank you very much.
Let me remind you that another poster session is scheduled, so if you are interested in one of the poster, you may also continue during the next break. Ladies and gentlemen, let me tell you that recently I was told that the European technology ecosystem has flourished in the last decade. Then I thought, okay, so maybe here among us there are some academics who view themselves somewhen in a private sector. And that's why I think now it's time for some do's and don'ts on startups which aim to spin out from academia. Once again, Dr. Bartosz Wawrzynów, together with distinguished guests, this time about building investable startups within the academic landscape in Poland.
Thank you very much, Katarzyna. Ladies and gentlemen, I hope, well, I see the seats are filling up and your meal was up to your liking. I'm really excited about this upcoming panel out of many reasons. This is I think, the reason why I joined and started collaborating with the foundation, I think 5 years ago. And the fact that I really believe in canonical, transparent tech transfer, which is aided by people that understand how to create or work on a repeatable business model. And without further ado, I mean, it's my absolute pleasure to, this time wearing a different cap, not his professor's cap, but I'd like to welcome Jacek Jemielity, the president of Explorna Therapeutics, to the stage. So please choose a seat. Our second scientist turner entrepreneur will join us shortly online, but I'd like to, in no particular order, I'd like to welcome to the stage the people, the institutions, the teams that really work hard to get startups to a level where they're actually thriving. And if I could welcome Jan Haberman, partner at Creative Ventures, to the stage. And of course, last but not least, the man, the legend, Marcin Hejka, co-founder and general partner of OTB Ventures. Is Piotr Karstecki joining us? Is he online? Then. Okay. Phenomenal to see you then virtually. Ladies and gentlemen, the spirit is movens, the CEO and the creator of Scope Fluidics. And again, a canonical example of a scientist that chose the entrepreneurial path. So, Piotr, welcome to the stage. I think we can turn this mic off, because I have one here. Yeah, thank you. Just before we get into it, I need to have a full disclosure to the audience. So the Foundation for Polish Science is an institutional investor through an SPV FNP Ventures that I am employed in, into active funds that are operated by Credo and OTB here. So this is me doing the legal bit. But now, as we enter and try to get to the thick of creating sustainable spin-outs that within the academic landscape, I think we need to start with the players. And the question that's definitely on my mind is setting up a company. How does one even approach the idea of setting up a company or incorporating or entering in full speed the entrepreneurial path near the vicinity or within the academic campus? And me being Polish, I have to ask about any stigmas, dogmas, general perception that that you may have, but at the same time, the upsides of it. And I generally, I really want to ask Jacek and Piotr for their input into that. How do you guys, because you're seasoned entrepreneurs now, it's not that you just incorporated, and how do you see, in hindsight, how do you see this now? Well, how do you see the process of creating a venture at that stage, and how do you see it now? So Jacek, if you could start, that'll be great, and we could comment and keep the momentum rolling.
Sorry, could you repeat the question?
The question is, what was difficult, what was terrible, what was great when it comes to setting up and thinking of starting a company from academia?
Yes. So let me first say a few words to Piotr.
Okay.
Piotr, I would like to congratulate you. Very great success, because probably most of you know that Piotr, as a founder, Scope Fluidics, did a really big deal in our very difficult environment. And I'm quite jealous person, but I'm really proud of you, Piotr, and I would like to congratulate. So big applause to Piotr, please.
Yes, definitely.
Thank you, please direct the congratulations to the whole team. This was the work of 70 plus people, maybe 100 plus people over the many years that we developed the system. So, but thanks.
And it was quite difficult decision to start the company. I'm a person who is involved in development mRNA, and mRNA now is very popular because of COVID vaccines. And I started modification of messenger RNA 20 years ago, and at some point I start collaboration with Ugur Sahin and his team, who is founder of BioNTech, well-known company nowadays. And it was a really inspiring experience, and we licensed them our first important invention, which protects mRNA against premature degradation in cells. And I learned a lot of different things in this experience, but at some point I lost control on our invention, and I thought maybe it was possible to commercialize our next inventions in a different way, and that's why
convinced by several people. I saw Chris Kaplan on the audience, so he was one of them. With my colleagues from University of Warsaw and Medical University of Warsaw, we created startup with 2 pillars for modification. One is related to modification of technology, development of the technology, and the second, use this technology for innovative therapy. And I was very enthusiastic because it was something new after publishing more than 100 papers. I felt that I will start something new, but I found several really big difficulties, barriers, because at some point I needed expertise, but I didn't have it, and this expertise is necessary to create successful company. But what is the most important for startups? People, money? Not really. The most important thing is IP. Without IP, these guys probably wouldn't talk with you. And the critical moment is to find person who believe you and who wants to give you money to start very expensive journey, because in biotech companies are so expensive, they spend so much money, so you need to convince right person to give you money and to make your dream come true. And probably we will discuss later the critical things, but access for IP experts, access to lawyers, and access to educated people. Now this is one of very critical issue.
Thank you. I mean, Piotr, if you're with us, Jacek started this, so definitely, I mean, epic congrats on the M&A with Biorad acquiring Curiosity Diagnostics. It was super refreshing to see on Business Wire a Polish entity being acquired through the canonical channels by a huge conglomerate such as Biorad. So that's really, I mean, that's a driver to us all, showing that there is a method in this madness. But I mean, let's take that and let's if we could add on what Jacek is saying. What were the key drivers to get you to the table where PCR1 was as a platform was considered, and an LOI materialized from Biorad's side, rendering this deal that's an inspiration to us all. If we could.
So I'll start with disagreeing slightly with Jacek. I think that IP is fetished too much. And what I mean by that is that IP is surely a very important component, but it's just one of the components of the product, and it's workable. So maybe in biotech or pharma, IP is fundamental and would be listed as the first thing. But you're asking about what sold PCR1. Many things. We've been to some extent lucky and to some extent wise and flexible to target the PCR1 system to the very attractive, commercially attractive niche on a dynamically growing market. So of course, pandemic amplified the interest in molecular diagnostics, point of care, but point of care format of molecular diagnostic was growing since 2005-6 very rapidly. And PCR1 addresses the highly multiplex, so the ability to offer a very fast analysis, completely automated, inexpensive, so accessible, but also with a wide range of information, so targeting multiple detection of multiple genes at the same time. And this is where the margins on the market are highest. This is the most attractive and also most prospective segment of the market. So this was one component. The second one was what was really difficult, and I think in the context of discussing the chasm between academia and industry, this is very relevant. So this is what we don't see typically from academia. So this is manufacturability, this is quality assurance system, this is the IP and taking care of the IP portfolios. These are the legal aspects. This is financing. This is organization of the team, communication within the team. This is suppliers taking care of supply chains for the manufacturing of cost, etc. So the whole kind of industrial package that create a product that can be produced and sold. And this was difficult. It took us many years to learn how to do this with trial and error along the way. So this was, I would say that this is the second component. And the third one, probably, I don't know, surely super important is the team. And it was to a large extent the team that has solved the system. We had visits from multiple companies, very intense, and we had 30 or 40 people interacting directly with the, I don't know, 10 or 15 visitors, different kinds of specialists, and talking, on an equal level about all the aspects of the project. So this was really a huge value.
Fantastic. I mean, you mentioned words that I hear on occasions on VC meetups or accelerator meetups or demo days. Niche, unique value proposition, UX. I mean, these are startup terms. These are your bread and butter. But guys, really openly, let's start with a blank slate. I think there's a portion of the audience that has a misconception. What do VCs actually do? What they don't do? What they should do? And the canonical question is, how does a VC invest? But prior to that, how does a VC raise money or make money? I know that there's the two of you, and we can split, but these are the basic questions that I think we need to answer before we start hacking into problems that you further see in deep tech companies around academia, or you may not see them. But before we get into that, let's continue the intro of the actors or the players that are on stage. So I don't know if Marcin, you can chime in, start, and then Jan, please chime in.
So to answer this question, I would say that venture capital is the only effective way of funding innovations. Okay, so basically any company that is, if you take a look at top 10, top 50, top 100 most successful technology companies in any country, definitely in all mature markets or globally, you will realize that every single one of them was supported by venture investors. And looking at multiple options, I would say that there's no way to be truly successful based on grants. There's no way for a startup, even having significant IP, to get funded by banks. Actually, the only way to successfully grow innovation in a business based on innovations is venture capital. And this is what venture investors understand and know, and the role of venture investor, and this is what venture investors should be doing, is supporting founders of innovative companies in this journey. The huge advantage of venture investor is that every experienced venture investor probably saw tens or maybe more than 100 of companies that went through this journey, and good venture investors understand what can go well, what are the risks, how to address them. Each venture investor should be able to open doors to potential customers, partners to help with funding, etc. So basically, I would say this is the reason why there's no other effective way to grow any business based on technology to any meaningful success.
Bold, Jan, go on. I mean, the train is moving, right?
To keep the train moving in that case, there's not much I could add to Marcin, apart of maybe saying what VCs don't do or should not be doing. And there was some discussion between the founders, what is the core thing of a company out of academia or early tech company, whether IP is more important or whether people are more important. I would say it's obviously all the combination, but VCs very rarely invest just in IP because they don't assume and they don't take over the control of the company, and then they don't build business on behalf of the company. So the company that is supposed to get venture capital funded needs to actually have a business idea at least, and understanding of the problem that the company wants to solve commercially. And having someone in the team who has the drive and the ability to build that business as well. So VCs are there to help, but they don't take daily control, and they don't actually perform that on behalf of the company. And if you look at it, it's very logical because our funds typically have tens of investments, and there's just a handful of people. So our firm is 10 people all together, and we manage 4 funds, and we have 74 companies in the portfolio across 4 funds. So we don't really have the even time or ability to be on daily basis active with those companies.
And then second part of the question, like how VCs make money, right?
Yep.
So first of all, I agree with Jan. IP in itself is not worth much, and no VC would invest in just IP. I think IP needs to have commercial potential. Needs to be, IP is great, but IP is only interesting if it can be turned into a product or service that is somehow addressing the needs of some group of customers who are willing to pay for it. And this is where VCs can help in commercializing. And how investors make money? By exiting, by selling those companies. And it's quite interesting because there are typically 3 possible ways of VCs or founders can exit the business successfully, and they are often described as 3 T's. And those are traction, technology, or team. So the most desired form of exit is driven by traction, where the company is able to scale its business to reach significant revenue and significant market share, and it's being basically acquired for typically quite substantial amounts of money by strategic buyers who are really buying revenue and market share on top of technology. And here we can talk about transactions, multi-billion-dollar even transactions based on that. The second category of exit is the second T, technology, where basically company can have very limited traction, but its technology is very relevant for a strategic buyer, and it can be applied. So in this case, basically the potential buyer is looking at revenue of the company that is being acquired only through perspective of this being a proof of concept, the proof confirming viability of the product. And in this case, multiples can be really much higher, but valuations are typically in the range of double-digit to low triple-digit $1 million. So basically, the rule of thumb is that every major US corporation, has certain threshold. It may be 200, maybe 250 at the largest corporations, at maybe 300 million level, where acquisition decisions are being made on business unit level. Anything above that needs to go to the board. So actually, if you take a look at valuations at which acquisitions because of technology are being acquired, what are the typical ranges? Those ranges typically from, let's say, 30, 40, 50, up to $250, $300 million, not really more. And finally, and this is really the fallback plan for investors and for founders is the sale because of the team, where basically in situations where the company fails to scale, when the company fails to prove the validity of its technology, it can still get acquired because of the quality of the team. And those kind of exits, typically can at best return the cost of of investment to investors, but are unlikely to produce any meaningful exit.
Let's work on the 3 T's. So what do we need to do? What do entrepreneurs operating already in academia need to do to, let's start with the team, right? Team composition, the dynamics of a team. Jacek, you have a team, right? Yeah. And you have a scientific team as well, right? Yeah. So you're kind of both. You have your science hat and your entrepreneurial hat. So what are the differences here? And what's the key driver of your commercial team?
Yeah, so of course I'm standing with one leg on the bright side and with the second on the dark side. And in both sides, the team is crucial. Of course, without team, I wouldn't be able to prove anything about my technology. But to build team, I need money. So, and to be honest, I'm not trying to offend you, but when we started our company, I met several VCs in Poland, and to be honest, the level of funding they offered is far not sufficient to start the team to prove biotechnology.
So for this reason, we have you guys that you represent a benchmark that I would love for Poland to have when it comes to the dynamics of investing in. But I really don't want to talk about money, but just Jacek to explain. OTB and Credo, they invest at a higher level. They co-invest, they do follow-ups. It's not a one-off transaction. And to tell you the very truth, true VCs don't operate in a one-off fashion because it's a journey of 2 sides, right? But apologies.
Mm-hmm. If I may just very quickly jump in. There's one thing that needs to be said, and it's not easy on a panel like this with a limited amount of time to explain all the basics and all the details, but same as with academic projects or with startups, there are tons of different breeds of VCs. And that's actually good for founders and companies that want to get financing as well, because it's important, the same way as VCs are picking the investment targets, the same way the founders should be picking the right investor and the right profile. And there are investors specializing either geographically or by stage of the development of the company or by the vertical. And everybody these days is able to get references on those investors. So the due diligence that investors are making on the companies should be the same way repeated by the companies who are seeking financing of the investors.
So in our case, we convinced Black Forest Fund, the fund from Luxembourg, but owned by Michał Sołowov to invest in us, and when after it was long journey to sign the investment agreement, and this is different story, but we finally were successful. Then we can start build a team, and for academic people from academia, it is quite easy, because in academia, sorry for this expression, we produce a lot of experts in the field. So in our case, our first team members were members of our scientific teams, because not all of them want to stay in academia. And it was a really huge synergy, because we wanted people, they wanted jobs, so we selected right people, because we know them very well. So we built team according to expertise we needed. And so when you have money in academia, to build team is relatively easy, but of course you need scientists, but you also need people for financing and for business development and many other expertise, and you need to find them on the market, and they are, sorry, scientists, they are more expensive than scientists.
Right. I promised. Well, I'm moderating this panel, so I don't want to take up space. But guys, later, if you could talk about vesting and ESOP options and other ways to remunerate a worker, then.
If I may, I can follow on exactly on what's been said, because the fact that you say it's easy to find people actually shows one of the important qualities that founders like you should have. And this is the ability to attract the right talent, because it's not easy for everyone, believe me. It's easier for the people who understand what they want to build and who are able to motivate people and who have some preferred access to academia or whatever group where you want to hire. And if you have it, you have much higher chances to succeed as a founder of a startup, which you apparently are a living proof that it works like this. On the other hand, then when you hire the team, as Bartek mentioned, you need the people that are working within the startup need to be motivated. So working in a startup is not for everyone. It takes some risk. Obviously, the environment is much less stable than in either mature corporations or in academia. So not everyone thrives in such environment, but there are people who benefit from the flexibility and from the excitement and constantly changing environment. And from the VC perspective, the good founders and good startup should be able to retain those people and help them to grow with the company. And the experience of VCs clearly shows that the best motivation for that kind of people is to let them also be motivated by the success of the company by making them co-owners by employing these stock option programs. And that perfectly aligns the interests of the people within the company with the commercial success of the company. And it aligns the interests of the investors as well, because then everyone is basically motivated financially by the success of the company.
I would add that ESOP is the most valuable currency on the planet. So if you think about it, ESOP is the only way for startups to attract talent from corporations. Otherwise, no one would be willing. Corporations will always be able to pay more. But there are so many examples of basically really highly paid corporate people leaving corporations just for much lower salary and joining startups because of ESOP. And once again, I think, you know, if we take a look at the list of the most successful entrepreneurs and financially in any developed country, absolute majority of them are people who made money because of shares or options in the successful technology companies.
To clarify to the audience, ESOP means employee stock option pool. It's basically a way of attracting talent, like Jan and Marcin said. Apart from a canonical salary, you receive options or shares, and you're a co-owner with time under certain conditions of a given company. And yes, I mean, it works in the market. So the third T, traction. How do I get traction, you know, from academic startups? How do you get traction? Do you have traction? Market traction. I mean, how do you attract through showing? You definitely have the technology. You definitely have the team. Where's the traction? How does the traction look at an academic perspective? How do you can foster it? How can you get the involvement of the market? Get more, seeing that, you know, you have this deep tech company in academia and big pharma companies saying, this is Jacek Jemiliti's company. We really want to collaborate with him. How do you network firstly to get that? You know,
I didn't catch the question, because for me it was quite easy again, because of my scientific background. So and my experience, and I would say some success story with BioNTech. So now, if our Explorna Therapeutics collaborate with more than 20 companies, and to start the collaboration is very easy, because they know our works, scientific works. So they know our achievement on these fields. So we proved them that we have something to offer, and it opened the doors.
So your scientific career, your merit,
yes,
was a gate opener. Piotr, which T are you? Your technology, right? Now let's. I don't know if I'm asking about curiosity or scope fluidics. Are you more interested? Are you more driving the technology side, or you know, traction, interest, the whole market engagement is more of a, you know, within your focus now?
So definitely, in what Marcin was describing, we were in the second category of technology. We had almost no traction. But the technology is very prospective. So in the hands of a large, capable company like Bio-Rad, this is a very nice prospect of large market share due to the technological advantage in the future. So technology. So we at Scope Fluidics, we focus on technology, definitely. That's where the fun is in organizing, you know, difficult, now it's fashionable to say deep tech, but generally technological projects. But I think that the traction is super important from the very beginning. So you have to think about the the product and be very humble, be very elastic in how you modify I mean, be ready to modify the functionality of the system of the product, so that it really suits the needs. There are many aspects of a product, and really from an academic perspective, you can have a super cute, super smart gadget that is great on, you know, on 10 aspects, but if it doesn't click on the additional 2, it might just not be a sellable product. So it's super important to do the hard homework and check what are all of the critical parameters of the product that would secure the sale. And this could be very unintuitive for people from academia, like for example, you know, the mode of servicing. I'm sitting in PCR1 now mentally. This will be different for different kinds of products. But if you have an electromechanical optical device that does something, it will break, and if it breaks, it has to be serviced. And this is, you know, I think that you have to think about when you develop, when you design the system, because servicing will incur cost, etc., and it will change the profitability of the product. So it might kill your otherwise ingenious technology as a product. So this is, I think, that's where the charm is, but also the challenge is that you have to add to the academic perspective, to the innovation, also the multiple kind of industrial perspectives to make a successful product.
Okay, thanks. I mean, really putting color on that. And do you think?
May I expand on that a little, because I feel like we might create an impression that as a new company, you have a possibility to decide whether you would be building a team or a technology or go for the direction. This is obviously not the case. Every company that I know is trying to build that traction because they come with a problem that they feel they have a great way to solve. The problem is big enough and they have a unique way how to solve it. But not all companies obviously succeed. Actually, in our space, in venture capital, the failure is much more likely than the success, and especially the outside success that everyone then reads in the media about, and that brings those millions to all people involved. But if things don't go that well on the traction side, the companies that are on the way able to build good technology still may have a chance to get some value of that. Just wanted to make that clear.
Thanks. That's, yeah, I mean, that's definitely putting the scale of success and the scale of failure. But guys, what is, I mean, let's imagine that, you know, Piotr and Jacek are starting from scratch. And a question to you, seasoned investors, is what is investable? And what shouldn't the people searching for, or even considering the financing roadmap, what should they do? But the don'ts. The don'ts are definitely the things that really annoy or really drive, you know, a distance between the investor side and the academic side. I mean, we also went through a process of due diligence, finding an investor. This is a whole issue, right?
Terrible story.
I mean, there are some standards. It's still for, you know.
Marcin, bring a better story.
I would say that you know, regarding due diligence, it all depends on the stage of development. I would say at early stage investors, let's say seed stage investors, pretty much the only due diligence they can do is to look the founder in the eye and ask themselves a question whether I believe, you know, this team can succeed. But regarding don'ts, like, you know, what there's one thing I believe is really super important for technology companies, and this is there's no such thing as local technology. Technology is either global or it's not technology. And the reality we are facing in our country, but it's in our region actually, more generally, is that our region is absolutely amazing in terms of talent, absolutely amazing place to build world-class technologies, but this region is not where you scale any business. And the reason is very simple. Like, you know, our country or our region is not an early adopting region, neither on the enterprise side nor the consumer side. So basically, if we take a look at structure of our economy, we see that economies locally are dominated by state. There are plenty of state-owned enterprises, and by definition, state-owned enterprises are not innovative companies. Or by subsidiaries of international companies who actually are not interested in searching and supporting local innovations, because innovations are coming from headquarters located somewhere else. And I would say that unfortunately, many companies are spending too much effort and too much time trying to scale locally in their local markets because it seems easier, it seems cheaper. You mentioned that actually hiring international commercial talent can be very expensive. But at the end of the day, it doesn't work. Because even if the company is able to win some customers locally, it doesn't help. Because those customers are typically not recognizable as references internationally. So ultimately, it's still impossible to scale the business. And local customers do not even serve as a reference. So basically, operating internationally requires basically starting from scratch. So the typical advice I have to companies who have truly unique technologies is to go international as fast as possible. And locally, if working with anyone locally, it should be someone who has a brand that will be recognized internationally. So in a way, Israel is a very good example how to use local talent to build success based on technologies. Israeli companies don't sell much in Israel. They go international from day one. And I would argue that actually absolute majority of Israeli talent is coming from this part of the world. We have equally good talent, but the market is much bigger. So we are punching below our weight for the time being.
So can I mention one real barrier for startup also?
This
very
brings us
important.
to a great point, because there'll be a time for questions from the audience, but I wanted to get a lightning round from you guys. So one-liners, snippets, that key point that you want to sell. Piotr, we haven't forgotten about you. Apologies for, I'm not seeing you directly, so I'm having trouble. There you go. There, I see you now. Okay, you started. Go.
So I wish to have a good funding agencies which fund R&D in Poland. And this is a great barrier. Now we have a really difficult time because we have some kind of funding gap. But even in the past, I have experience with FNP, with National Science Center. But when I moved to agencies which funding R&D, so I noted a lot of different approach, a lot of strange things, and this is really important for startup to have to start funding from public money, with support from venture capitals. But in our case, when we submitted application to one agency, and then we received a report from reviewers, and we learned that our project is not sufficiently innovative, right. And it was quite strange because I believe we have quite strange IPs, but I was even more surprised when I saw the results of this competition, and the winners were innovative production line for pierogi. We talked with Bożena Kamińska about this example, of course, and even more surprising, the innovative way to extract meat from pig's head.
Let's park at
and
that.
so, as you see, competition is very high.
What I'm taking from you is the cost of capital is very important, and the way you logically structure capital, whether it's equity-free, and the source of equity for financing is very key for the operation of the company. So that's my synopsis of that. Piotr, if you could give us a one-liner of to future entrepreneurs or future people that actually really want to go on this journey.
It's going to be a one-liner. I would say, to me, especially speaking at the Foundation for Polish Science conference, so speaking to scientists, I think it's important to. I mean, at least I have the impression that science is a little bit asleep. In the sense that we in academia we go up these crystal towers of our toys and models, etc., and we don't look enough at very practical problems. And Toyota proposed the 5 why questions to solve problems in industry. It's also known in science. It's enough to ask 2 or 3 or 4 times why to get to really to the edge of understanding of the world. So why not take practical problems? Why not take problems of storing energy, of I don't know, curing that disease or any other practical problem, as opposed to being locked in our routine kind of set of models and tools that we use in science. This is, I think, my biggest dream for a change in science, because I think science would benefit actually from the level of science, but it would also bridge science to practical applications. And the rest is, it depends on whether you have the taste for the venture or not, but it can be a lot of fun. Because once you progressively solve the problems with funding, organization, you build a big venture that can be much more exciting and bigger than what can typically be done in science. So I think it's a worthwhile effort.
So I'm going to take the relevance of finding addressing issues that really have a tangible problem and actually addressing them while being fully engaged and having fun, which is also important because, like you guys said, this is not an easy venture. And Jan, I mean, you sold me a long time ago on founder market fit, so I'm going to take that, and I love that. But something else, something that just pops in your head.
Actually, that was exactly what I just want to mention. I'm not sure that buzzword from the VC world is familiar to you guys, but venture capital investors look at patterns. We try to recognize what we've seen before, as Marcin mentioned, what worked and what didn't work. And we have a name for that when we talk to a founder, whether that person has a founder market fit. And what that's supposed to mean if we believe that the founder knows what the problem is, what problem is he or she is solving, and what it takes for this problem to be solved and to commercialize the product or technology that the company has. And this is a totally different mindset from the mindset that researchers in academia have. And as much as the technology and the research is a very important part of the company, and deep tech company without technology cannot be built, this is not sufficient for getting financed by venture capital. Getting financed by venture capital is also very different from applying for the grants, because we look at different criteria. We don't objectively measure that. Obviously, we try to apply some objective measures, but there's still a lot of subjective. And there we use those patterns and pattern recognition, how we try to evaluate. So I would encourage anyone considering that venture capital is a good form of financing, think about the business model and commercialization from day one, and try to adjust everything in the company in order to be able to commercialize. This is the best way how to attract financing from someone like Credo.
Marcin, if you could.
Yeah, I think probably not much to add. I think looking for looking for a real problem that some well-defined group of customers is willing to pay for. That is a problem that is preferably global problem or international problem, definitely not local. That is the good way to attract VCs.
Right. So, ladies and gentlemen, we have time, and we openly ask if there are any questions in the audience to please address them. I see Professor Kamińska. I don't know if there are any microphones operational. I can...
I totally disagree with diagnosis of science and scientific approach by Professor Garstecki. First of all, science is a fun. We researchers address fundamental issues. We try to understand and cure diseases, and this is not the area limited exclusively for the companies. I actually believe, and this is my experience, that we make discoveries, and then we have to provide the product. So actually the applied science is less attractive because we need to find the drug formulation, the delivery way, we have to make a product, we have to think about packing, transferring, and so on. So I actually believe that this is, of course, could be attractive for some people, and this is some kind of venture, but it's totally wrong to believe that science is not an adventure. I believe that many of us in our science address really breakthrough issues, and this is actually very important. And if someone would like to make a product, go with something, it is of course worthwhile, and society needs the product, not the great idea. But you cannot say that this is more worthwhile. This is just different activity, and you need people of different attitude to follow this pathway, because this is the different way of looking for science. It's scientific.
I don't know, Piotr, you want to take this, or should I?
Can I reply? So if I have the voice, I was misunderstood. So of course I'm not. I think that there is a lot of great science going on, but what I'm saying is that a big fraction of the scientific activity is somewhat lazy intellectually, in the sense that we tend to stay in the safe position of the tools that we know without going back to the street and being inspired by bigger problems. And that's all I'm saying. I'm not trying to influence anyone to get interested in packing or confection. I think that science is great. I'm just saying that good, I don't know, 2/3 of scientific activity could be greatly refreshed to the benefit of the scientific discovery if we took a step back from our tools and models and thought about bigger problems and how we can solve them as well.
Okay, thank you. Maciej.
Thank you very much for the nice talk. I mean, it is not common to sit in an academic position, academic conference, and hear the voice of a venture capitalist and investors. So it was very nice today to hear a different voice from a person like me that works in academia. So the question that Professor Maria just said, and the answer that she received, actually, it was the opening of my question. So we, as academic persons, we like to do our stuff. We like to spend our time in the labs, a little time back in the laptops and writing our articles. Sometimes we come up with a very nice idea that can become an IP and later a very attractive technology, but they are like fetus. So I will leave some fetus around here and there. They are not even babies that can be raised and become a company later. It would be very nice, for example, if someone come and find those fetuses, put them in an incubator, raise them to be a baby, and then make a successful company out of it. But it is not actually what the background of my experience can lead to. So, but then we hear this very interesting media from a venture capitalist or investors or the nice environment of the startups. We hear those things, and we are a little bit motivated to go through it, to go to that part. However, for example, today, I think I heard it twice from you that, for example, this is one of the things that a venture capitalist would not do, is making a company for a scientist, right? This is not that you are willing to do. This is my job to find my team. This is my job to write my business plan and to go into the mouth of the devil myself. You will just help me a little. You just take my hand and drag me toward it. But I mean, so this is what has been said today. Then you would take a look at the success rate of of a startup around the world, and you will see that 90% of those startups fails. For example, you have some very good. I will close.
Sorry,
my question.
Really
Yes.
Is there a question there? Let's form a question.
So, would it be better if you, as a venture capitalist or angel investor, or for example an accelerator, just make my team just govern the company that can become out of that technology to higher this success rate, and let me be at my lab. Let me be spending my time in the lab to come up with the next idea. Thank you.
So I assume I might be the addressee of that question, so let me very quickly react. There are some institutions trying for that, but you said that yourself, that very few of those are successful. And the reason why very few are successful, because this is damn hard to do it. Aligned motivation in such setup is extremely difficult. And that's just not how VCs like us operate, because we don't have the capability and we don't believe we would be successful in this model.
Can I
really quickly, of course.
So from my perspective, the development of technology without inventors is impossible. I like this comparison with the child. So at university, we care about our child in primary school. Startup is a secondary school, and the goal is to send our child to high school.
Okay, with that said, yes, we believe in higher education because it gets us into university. But I think the president of the Foundation for Polish Science is entitled to a question.
I'm not so sure if I am entitled. I would like to just ask Piotr and also other. Is it the only way to be successful to have help of VC or money from VC? So Piotr, did in your career, in your success, do you have help of the VC?
Well, I think that it's critical that you do not rely on grants only. I think that grants are very good maybe at the early stage. They might be helpful later, but they're also quite dangerous, both because of the bureaucratic risk of reporting them and lack of flexibility, but also the lack of discipline in the sense of someone watching how well you spend. So it's absolutely necessary to have private funding, and I agree that VC is probably the predominant hand here. In Poland, we also have this growing sector of technology companies growing, being funded from the public market, which I guess is somewhat unusual looking wider because the long-term technological projects are not super compatible with the stock exchange. But nonetheless, there is a number of them on the market. We've also been on the market since 2017 on the stock market. So it is a possibility to go with public funding. Public meaning stock market, not grants. Does it answer your question, Maciej?
No, Maciej was asking if you personally ever divulged into the honeypot of venture capital as you were building businesses, but I don't know if we still have time to continue this. Yes, if Piotr, if you can directly answer that.
We had private investment because we IPO'd, so we did.
Okay, thank you. I think we've come to an end. I really appreciate your time and your input. And Piotr, thank you very much for two things: being responsible and social distancing. It's a quality that's becoming less frequent.
It would be so much nicer to be with you.
Well, of course, but at the same time, thank you for being with us in the best way that was provided. Gentlemen, thank you to the brethren from sciences to entrepreneurs. I wish you all the best, Jacek. Full speed ahead, and I really hope to see more presence of VCs at conferences like this because they share a completely different perspective, not compatible to everyone, but this is the whole point of this conversation and this conference, I think, to network and to have different ideas. So, thank you very much. I think we can conclude and we can depart from the stage.
That was a lively discussion, wasn't it? For me, it's time to say thank you. It was a great pleasure. But of course, our conference is still going on. From now on, my colleague from the FNP, Justyna Motrenko, will take care of you. Justyna, over to you.
Thank you very much, Kasia, for taking care of all of us during the first part of the conference. And now we have had another part of the poster session. So please let me draw attention to the posters number 81 to 130, whose authors maybe you would like to find a new seat for this very last session today to have a change of perspective.
Dodałem 5. raz jest. Chyba ta magia białych kart, dokładnie.
I know that there are still some interesting conversations going on in our poster area, but I would kindly invite you to take your seats, and I promise you that this last session will be also very interesting, related to different topics, different research specialties. One of the tasks that is often taken up by scientists is to react to the most current social needs. Undoubtedly, COVID-19 has been the scientific challenge of the past 3 years. FNP laureates were also actively working on COVID-related topics, and now is the time for them to present their results. I'd like to invite my colleague, Dr. Radosław Kwapiszewski, lead project manager in our technology transfer support section, to chair this session.
Good afternoon. Is it working? Yes, it's working. Okay, good afternoon, everyone. It's a big pleasure to be here and to meet with all of you offline, finally. And before we start a section and we start presentation with our speakers, please let me share one thought with you. If you ask me why we need scientists, what's their role in society, I would say that they are all together. When we put them together, they are like a library, like a living, alive library. And even if we need some information, we can find them in this library. If there is something unexpectedly happens or something unexpectedly occurs, then, and there is no answer in the library, no one else but scientists, they are the one who can find a solution, who can take up a challenge and give information, more really reliable information. And COVID-19 pandemic is a great example of how much we rely on scientists, how much we rely on verified knowledge. And many scientists worldwide have already changed their plans, their research agendas just to meet social needs. And I'm really happy that some of these scientists are today among us. 2 of them are present online and 2 of them present in person. And they are going to share their results and their conclusions with us. What is even more exciting, that the topics they present are not from the same life science area. And I'm really, really curious about the session and your questions. So please welcome our first speaker, Professor Agnieszka Michota-Kamińska from Institute of Physical Chemistry, Polish Academy of Sciences. Today she is in the United States, but is going to join us online. Professor, can you hear us?
Yes, good afternoon. I hear, but I can't see my presentation of another screen. Okay, now I suppose that should work.
It's there. So please, the floor is yours.
Good afternoon, ladies and gentlemen. It is my great pleasure to participate in today's meeting, in today's session, even if it's carried out remotely to my abroad state. During the Team Tech project, we have developed the SERS, which means Surface Enhanced Raman Scattering Device, for the detection and identification of pathogens, bacteria and viruses, including SARS-CoV-2. We have developed the SERS-based sensor for which challenges the current method of pathogens bacteria in terms of selectivity, sensitivity, and time of analysis. So, before I move to the main research subject of my today talk, to COVID-19 research results, I must say a few words about our nanoplasmonic structures, which are the heart of our cells technique, of our methodology. And as we started our project with bacterial detection research tasks, and additionally, in many cases, in many purposes, we need to differentiate bacteria and viruses. I will also show our success in detection of bacteria, only one example. So, we have designed, fabricated, and patented several different cells active nanostructures, several different nanoplasmonic substrates with high sensitivity, stability, reproducibility of recorded signals, and we combine them with microfluidic devices and developed portable Raman spectrometer for SARS-based analysis for point-of-care SARS application in detection bacteria and also in COVID-19. And this is only one example of our success in detection of bacteria, which I show. Sometimes it's difficult to differentiate between bacteria and viruses. You have to know this knowledge, and this is an example, as we have developed the optical sensor for the differentiation, for the analysis, for the detection of human venereal pathogens. Which, such as Neisseria, Haemophilus, Mycoplasma, as a novel approach for the gonorrhea or other sexually transmitted diseases analysis, and we are able not only perform bilateral differentiation into infected and non-infected sample, but also we are able to perform identification of particular bacterium at both strain and species level. So this sensor possesses impressive sensitivity, selectivity, is easy to use without pre-cultivation, enrichment, labeling of pathogens with time of analysis below 15 minutes, and it's practically easy to use, easy to adopt. Now I move to the main subject of my talk, to COVID-19. COVID-19 diagnosis. The pandemic of COVID-19 caused, of course, the damage, burden the economy and healthcare around the world. And there are, of course, method of COVID-19 diagnosing, including PCR, which, of course, this method is sensitive and selective, but also has some limitation, like may give the false positive, false negative results, is expensive, need expensive reagents, is time consuming, and the time of analysis is in hours and sometimes even day. And therefore, the most common immunological test, which based on the detection of antigens against SARS-CoV-2 are not good for early detection of COVID-19. Therefore, there is still need to develop very novel method, fast and cheap for detection and identification of pathogens. So, in our research work, we have combined the cells-based approach with chemometric analysis to establish the biochemical fingerprint of SARS-CoV-2 infected human samples. We have studied saliva and nasopharyngeal swab. The procedure of SARS-CoV-2 detection consists on clinical sample collection, the deposition onto cells-active support, cells measurement, and chemometric analysis. Numerical methods applied for our cells data facilitate the spectroscopic identification of viral specific molecules, molecular changes in quality and quantity of many proteins or other specific biomarkers, which reflect the immune system activation and disorder upon SARS-CoV-2 infection. As we may see, any differences between healthy and infected samples are reflected in SARS spectra and extracted by chemometric analysis. All SARS data induced to the PCA chemometric were nicely divided, as we can see, on two groups, CoV plus and CoV minus. We have found that the saliva biochemical composition of SARS-CoV-2 infected samples is manifested by the increased concentration of many proteins, such like ferritin, methionine, and as well as immunomarkers like immunoglobulins and interleukins. We have, for example, found that neopterin, its novel SARS-CoV to biomarkers. Neopterin is small molecules, pteridine derivatives produced by the macrophages in our organism, and its increased concentration appear upon infection. So this slide shows the receive sensitivity, selectivity, and specificity of our research-based method for SARS-CoV-2 detection. And so at the current stage, our device enables fast and non-invasive sample collection, time of analysis below 15 minutes, and up to 90% of accuracy of classification for saliva sample, which is the best working fluid in SARS approaches in comparison to tested nasopharyngeal swab. I would like to highlight that we have developed first in the world SARS database of pathogens. The library of pathogens currently consists on more than 150 species and fingerprint, SARS fingerprint of SARS-CoV-2 infected human fluid, blood, saliva, and nasopharyngeal swab. We have also developed the algorithm, the software for which enable to search information of bacteria, instrumentation, measurement conditions, and it also enables the classification of particular bacterium based on the developed, established algorithm. This slide shows how our setup works after uploading the unknown spectrum of our pathogens. The software founds, according to the algorithm, the most likely pathogen, and so highlighted it on the list on the pathogens which exist in our library. This last slide shows our forming device. So you have started on May this year the formal procedures to establish Formi technology company with participation of our institute as a shareholder, and we hope you're able to offer the products and analytical services, including SARS-CoV-2 identification. And this is the summary of practical application of our research work conducted by the Team Tech project. And I would like to thank my group members for the hard work, Foundation for the financial support, and of course, I thank you very much for your attention.
Thank you very much, Professor Michała-Kamińska, for your presentation. We are almost in time, and still there is time for questions from the audience. So are there any questions to Professor Kamińska? Okay, if not at the moment. I have a quick question. Which compound or which part of bacteria or virus is detected by your method?
It's a very interesting question. In the case of viruses, we detect the components of cell wall because the signal from SARS generally is dominated by the molecular species that are in proximity to the metallic nanostructures. Therefore, we detect protein, cell surface, antigens, generally compounds of the cell wall, and cellular membranes. But in the case of viruses, we of course do not catch the virus on the cell support, put them on the our nanostructure. Therefore, in this case, our cells technique is used to monitor, to find the biochemical changes which occur upon infection in body fluids. We find there are, of course, we can also catch and monitor antibodies against the SARS-CoV-2. But at this, what I say, what's the we generally measure the biochemical composition of fluid upon SARS-CoV-2 infection manifested by appearing in the cell spectrum of many specific biomolecules upon immune system activation disorder.
Okay, thank you very much. Are there any questions from the audience? Yes, there are plenty of them. Okay, so could you please pass the microphone?
Hi, here. Can you really distinguish SARS-CoV-2 from other respiratory viruses? Since you look at very indirect proxies, is it really SARS-CoV-2 specific?
Professor Michalowski, can you hear that question, or should I repeat it?
If I please repeat.
Yeah, the question is about how can you distinguish the SARS-CoV-2 COVID cells and other viruses like for example, influenza. Yeah.
Yeah. So far we were focused. It's complex problem. Therefore, so far we have focused on them because we working on them. In those years, we have generally problem. We find another virus sample, clinical sample infected by another virus like influenza. There was problem. We cannot find such sample even in many hospitals. It's another issue. But so at this moment, we do not working on such issue because we are working. I show the experiment, shown our data. It shows the differences in biochemical composition the fluids after infection. But another path of our work is selective catching, immobilization of viruses using antibodies, specific antibodies. And in this case, of course, we are able to. I hope you were able to differentiate, but it's not direct. It will be not direct technique, but using antibody specific to the different viruses.
So
such as we adopt also for bacteria.
There's one last question, comment from Professor Żylicz.
The question is about the accuracy of this technology. So you have, let's say, more than 95% chance that you correctly distinguish between COVID positive and COVID negative case.
We have tested saliva nasopharyngeal swab, and for saliva, the best accuracy of diagnosis was from 85 to 90% between bilateral differentiation, infected or non-infected. But of course, it's a complex problem. But it's for saliva, we receive so far such accuracy of diagnosis, up to 90%.
Okay, thank you. Okay, so very quick last question. So I know you have some patent application already released. So where are you now on the path to commercialization with your tests?
So as I mentioned, we have started the formal procedures to establish our forming technology company. We have a lot of patent, and we also will finalize international patent, which in the concern the COVID-19 detection. I suppose that So at this moment, we have a lot of products like spectrometers, special design spectrometers for search-based point of care diagnosis of bacteria and viruses. Of course, in the aspect of bacteria identification, we have started 4 years ago this subject, and and we have a bigger achievement in this area, but also COVID-19 diagnosis is also our issue, our investigation and further development.
Thank you very much, Professor Michota Kamińska, for your presentation. Thank you very much for the questions from the audience. Let's thank our speaker. And now it's time for our second speaker, Professor Justyna Olko from University of Warsaw.
Good afternoon. I'm going to talk about the results of our research on the impact of COVID-19 pandemic on ethnic minorities and indigenous people, research that was funded by the Foundation for Polish Science within our team program project. Something unexpected happened. I'm really sorry, I don't know why it went to the end. Can you restart my presentation so it starts from the beginning, please? Thank you. The COVID-19 pandemic has had devastating effects globally on many aspects of life, but the effects were especially pronounced among the indigenous people. The pandemic has virtually wiped out some ethnic groups like the Juma people in Brazil. The last representative of this tribe has died from COVID-19. And with some information, pieces of information I'm going to show right now, I'm using the data from the US CDC statistics because they are actually the only country that's registering ethnic affiliations at also with regard to healthcare. This is legally prohibited in the European Union. So even if these classifications might seem racist to us, they are actually useful for looking at vulnerability of certain ethnic groups. And in this chart, you can see that Native Americans and Alaskan Natives actually had the highest death rate from the pandemic, similar to the Hispanic people, mainly immigrants from Latin America, so also indigenous people. And during the pandemic, the life expectancy in the US has dropped to the lowest level since 1996. Between 2019 and 2021, this drop was almost 2 years. But in the case of Native Americans, as you can see on this diagram, it was, I'm sorry, this is not very precise. The drop was 6.6 years. And this drop has basically erased the equivalent of over 4 decades of American life expectancy gain, basically causing the situation that an average Native American is supposed to live as long as an average American in 1944. And this is obviously caused by a number of factors. If you look at these the maps that show the situation of Arizona that has a large share of indigenous population in the United States, basically the level of mortality in the Navajo County and Apache County corresponds with the level of poverty and marginalization. And this is not caused by the lack of protective behaviors among the indigenous people. In fact, the Native Americans have the highest level of vaccination comparable to the people of Asian origin, as you can see on this diagram that shows fully vaccinated people. And the bottom lines belong to Afro-Americans and people classified as white. Obviously, the attitudes of Republicans have greatly contributed to this effect. We have found similar effects for the indigenous people in Mexico. This was the area where we carried out some of our COVID-related research. We analyzed raw data published by the State Bureau of Statistics, and we found out that while the overall level of mortality, COVID-related mortality for people who were tested for COVID, and we know that these were very low levels, was roughly 9%. For the indigenous people, the mortality was 17%. And this was confirmed by other studies that showed that was the native people had 64% higher mortality rate, and this was actually twice as big with patients who died outside hospital. The goals of our COVID-related research in the project titled Language as a Cure, that was co-directed by Professor Michał Bilewicz from the Faculty of Psychology of the University of Warsaw, focused on different kinds of disparities that affected ethnic minorities during the pandemic, such as access to healthcare and effective communication. We looked at the impact on their psychological health and their economic situation, on the minority language use, on stigma and discrimination these people experienced, how they behaved during the pandemic, if these behaviors differed from the mainstream population, what was the impact of earlier traumas and marginalization to which they were subject, if their sense of identity was somehow associated to their behavior and also to the levels of perceived threat and anxiety, on social trust, and also on media discourses. And all this research during the COVID pandemic was carried out online through a number of surveys that were advertised via Facebook. And this is in this table, you can see our sample. The research was carried out in 3 countries. In Poland, it was in Podbeskidzie, in the Silesia, Kaszubia, Łemkowina, and you can see the number of people who actually, within our survey, identified as ethnic minorities. These are these blue areas, because some of the respondents identify themselves exclusively as Poles, and the same concerns Mexico, where we have a general sample, and within this sample, the majority were people who self-identified as indigenous. We also carried out survey in Calabria, where there are a number of migrants and also ethnic minorities. One of the areas where we focused most was Silesia, the region that was labeled to be Polish Lombardy. And as we all remember, during the first phase of pandemic, the Silesian people were presented and largely perceived as a threat because of the surge of cases that were associated with coal mines. And in fact, this level of perceived stigma, high level of perceived stigma, was confirmed by our study because almost 70% of Silesians, and as you can see here in the lower line, they thought that they felt that they were treated as a danger, as a peril during the pandemic, as confirmed to almost 7% among the Kashubs and almost 5% among the Wemkos. Also, Silesians felt that they were treated worse in a worse way during the pandemic because they were identified as Silesians. 44%, as confirmed to almost 5% of Kashups and an insignificant number of Wemkos. However, when we compare their protective behaviors, such as, for example, wearing masks during the first phase of the pandemic, the protective behaviors of ethnic minorities were much higher than those of, significantly higher than those of the outgroup, the majority outgroup. And this was not just the case of Silesians who were stigmatized and were sort of put in a spotlight. The same effect we found among the indigenous groups in Mexico, with regard to protective behaviors. Here you can see differences between indigenous and non-indigenous populations, and the level of resilience. Moreover, it has been demonstrated, and you could probably saw it yourselves, that men tended to respect restrictions in a lesser degree than women during the pandemic. And they were more prone to accepting conspiracy theories. So according to the study by Galasso et al., women are more likely to perceive the pandemic as a very serious threat problem and to agree and comply with restraining measures. However, as the analysis directed by Joanna Maryniak, who is present here in this room, showed, this maladaptive gender difference is actually smaller in ethnic minorities. And we've been able to show this based on research with Silesian, Kashups, and indigenous groups in Mexico. And here you can see the results showing that the differences between male and females with regard to protective behaviors and perceiving COVID as a threat, were much smaller. So the social cohesion and sort of like collaboration networks between ethnic minorities are different, and it's actually provides some adaptive effects during health crisis. However, in Mexico, we also could confirm that indigenous people felt discriminated with regard to the access of reliable information about the pandemic. As many as 44% perceived this sort of discrimination, and also over 25% of the indigenous people thought that the physicians took poorer care of them because they were members of indigenous communities. At the same time, they perceived similarity between COVID-19 and colonial epidemics, as well as Spanish flu. And colonial epidemics basically erased 90% of indigenous population during the first 150 years of colonization. So this is important source of historical trauma and historical memory among the indigenous people in Mexico. And almost 80% of people perceived the similarity to the colonial epidemics, and over 60% to Spanish flu. Why I'm saying about this? Because we were able also to confirm a re-traumatization mechanism during the pandemic, basically that ethnic minority groups that were subject to different forms of COVID-19 related discrimination also had a higher level of general anxiety disorder, but the mediator in this relationship was their sense of historical trauma and the presence of historical trauma symptoms. This effect was found to be significant among the Kashups, who were not very strongly stigmatized during the pandemic as compared to the Silesians, among Silesians, and the indigenous people of Mexico, where this effect was the strongest. On this diagram, you can see the full mediation analysis, where historical trauma and the symptoms of trauma mediate between discrimination and ill-being, the general anxiety disorder. And the same effect we've been able to show for the indigenous people in Mexico. Obviously, for the indigenous people, containing the pathogen is not enough to fight the deadly pandemic, and it's not enough to overcome the effects of the pandemic. One of the strategies in which we got engaged was information campaign in the indigenous languages of Mexico in 4 different regions. We organized a campaign to provide materials for the local people, and also collaborating with the local health system and distributing the information in the local variant of Nahuatl, as you can see here. Also, some kind of posters related to other health problems such as diabetes. We also have produced, collaboratively with the minorities, an anti-stigmatization package resulting from our project that combines the effects of research with very practical recommendations around historical trauma, discrimination, media and communication, job market, language use and policy. And this package is actually being now in the process of being published in Polish, Kashubian, Łemko, Silesian, and Ukrainian. There is still much to be done to erase profound inequalities between the mainstream society and ethnic minorities that are one of the that constitute an important source of their vulnerability during such crisis as the COVID-19 pandemic. Thank you for your attention.
Thank you very much. Thank you very much for your wonderful presentation. Are there any questions from the audience? Yes, there's one.
Actually, I have two. Thank you very much. I'm completely, this is completely not my domain, so I'll be looking at this from the immunologically sort of relevant mechanism size of view. So I wonder, with the vulnerability of these populations, how homogeneous are these? Because the populations, because immunologically, the more diverse the populations are at genetic level, the more protection that will give them. So I wonder whether there is something, because they, sort of endemic. That's the first question. And the other question that I have, if that's okay, so the difference between the roles, like the female and male sort of understanding of the rules and sticking to the rules. I wonder whether there is any correlation with the sort of stereotypic gender roles in these. Is that the same? Like the roles, do you know what I mean? Like whether there is a difference in those populations, so there's less of a.
Yes. Thank you for your question. So the fact that these populations, ethnic minorities, are isolated or might be isolated with results in their genetic, like very specific genetic profile, is a myth to a large degree, because obviously they are in contact with like linguistically and culturally, also genetically, there is a lot of contact going on. You probably heard about the so-called Kashubian gene, right? For example, yeah. But obviously this is different, and we cannot really say that Kashub, Silesian, or Łemkos are genetically very much different than the population outside or the dominant population, because they live interspersed. It's more the question of identity. Who considers to be a member of minority and behaves like a member of minority. So this was the primary criterion for us. And if you look at the population of Mexico, there are not very many genetic population, genetic differences maybe between the white elite, the so-called white elite, right, that lives in Mexico City, maybe, and the rural populations. But generally, it's not a question of genetics, it's a question of identity and ways of life. And the second question is about the cultural differences in male and female roles. Yes, and I think one of the reasons, Joanna is perhaps the better person to ask about it, because this was her idea to investigate it and she pursued this topic. But one of the factors we consider is so-called toxic masculinity. And this has different implications. But what's important for us is that the way men and women in traditional communities collaborate with each other are different. This is more collaborative, like I would say even more equitable ways of dealing with different aspects of life and doing things in community, not individually. And one of the reasons of this bigger protective behaviors among indigenous groups is that for them, and this is pretty salient in our research, I just didn't have time to talk about this, it's not just the concern about your individual life and a threat to your life. It's a threat to the community, your family members and your community, and the threat of cultural survival, not only biological, but also cultural survival of your entire community. So I think that these are very important factors to be considered. And these are really, I would say, certain adaptive factors in this kind of crisis.
Okay. Yeah, I hope it's a quick question, but how did the pandemic influence the use of endangered minority languages? So this population are stronger because of pandemic, or they are weakened?
Thank you. Yes. So obviously, we assume in the beginning that the pandemic will be devastating for different endangered languages because it will wipe out the elders, who are those who transmit the linguistic knowledge and the cultural knowledge. It's a very common pattern that children in minority communities do not learn their heritage language from their parents, but from their grandparents. So this was the vulnerable group. We thought, yes, this will be the immediate effect. And also some groups like the Juma people, for example, and they were not the only one, were really affected by high mortality. However, when we measured the declared use in different domains of life before the pandemic and during the pandemic, across our sample, we didn't really notice difference. There was no big drop in the usage of these languages. There were some differences, but for example, the average levels of language use were the same among indigenous people in Mexico from different groups and the WEMCOS. We think that the positive factor was the fact that they had to be close in their households, co-living with their different generations. People came back from the cities to their home communities, so they had more opportunities to practice their languages, for example, with their grandparents. So this was a paradoxical effect of pandemic, which was actually not really negative. Also, they used social media very much to promote the use and communication in endangered languages. So this was another positive side of the pandemic, that they really got digitally activated.
Yeah, thank you very much for the comment. We are a little bit out of time, but this discussion really fascinates me. I apologize. So our third speaker is Dr. Alicja Chmielewska from University of Gdańsk, and we are going to connect with her online. I hope she's there.
None?
Yeah, can you hear us properly?
Yes.
Yeah, okay, so no time to lose, please. The floor is yours.
Okay, good afternoon. Yes, I will tell you about the use of SARS-CoV-2 pseudoparticles that we used to test some chosen antiviral strategies against COVID-19. And this work was done in the Department of Molecular Biology of Viruses at Intercollegiate Faculty of Biotechnology, University of Gdańsk, and Medical University of Gdańsk. So, okay. I changed the slide, but it doesn't change. Yes. Okay. I was the PI of the project Powroty Reintegration, and that was about the characterization of interferon-induced transmembrane proteins, IFIT-Ms, as antiviral strategies. And in this main project, we used two viral models, hepatitis C virus, HCV, and tick-borne encephalitis virus, TBEV. And our proteins of interest, IFITM1, IFITM2, and IFITM3, they are cellular proteins that have antiviral functions. They are stimulated, their expression is stimulated by interferon during the virus infection. And they block the entry of viruses to the cells. And it was described for some viruses before. It was not described for TBEV. So we were interested in this and also interested to see the mechanism of this inhibition. Okay. And I think that the most interesting finding from this first part of the project was that first we showed that by generation of stable cell lines overexpressing IFITM proteins, but also by generation a panel of Cas generated knockout cell lines, that all the three proteins IFATM 1, 2, and 3, they potently inhibit TBEV virus. But on the other hand, we showed that TBEV virus can escape this inhibition. On the right, you can see the graph in which you can see two modes of transmission of viruses. So viruses can go out of the cell and infect another cell from extracellular space, but they can also go directly from cell to cell without leaving to the extracellular space. And we showed that while TBEV virus is going directly from cell to cell, it can majorly escape the antiviral activity of IFITM proteins, which was not shown before for these proteins, and it was published this year in Journal of Virology. So then I managed to get the prolongation for the grant for COVID-19 research. And it was quite a small grant because it was 1 year prolongation and financing actually work of one person in the lab. So it was done mainly to implement a system for research for SARS-CoV-2 virus. And because, as you know, this virus is highly infective and it's respiratory, so this is a high containment virus. So you need to really have special measures and safety rules in the laboratory where you want to investigate infectious virus. So not many laboratories can do this. So my idea in this project was to implement a safer system that could be used at once when we didn't have other possibility. Okay, so one of the idea was to use a milder, let's say, kind of coronavirus. This is NL63 virus, so it's the same family than SARS-CoV-2, but and has some similar properties, like it is using the same receptor, for example. So we made some, we amplified this virus, and we made some basic infection experiments and assays. And the next system that we tried was the system based on SARS-CoV-2 pseudoparticles, and this is in fact the system that we decided to work with later. And so pseudoparticles are particles that are actually lentiviral particles. So they have a capsid that is the capsid of HIV-1 virus. And outer of this capsid, there is a membrane, and in the membrane of the pseudoparticles, there are embedded S proteins, spike proteins. These are proteins, so surface proteins of SARS-CoV-2 virus. And pseudoparticles are generated by transfection of eukaryotic cells, and then these pseudoparticles are produced and secreted to the medium. And you can use pseudoparticles as a system that mimics the entry step of SARS-CoV-2 infection, so mainly the binding to the receptor and then the entry to the cytoplasm. Yes, so the major part of the project was actually optimization of the protocol, adjusting it to our laboratory, to our reagents, choosing recipient cells, and choosing the methods of detection. So, for example, we tested two reporter proteins, that is green protein, that is green fluorescent protein, and also a reporter protein luciferase that enables us to detect chemiluminescence. And we use the latter one for the testing because for convenience, because it allows us to test numerous samples in an automatic readout. So this was quite a major part of the project. And then we used the optimized system for the assays. So first of all, pseudoparticles are very good for neutralization assays. So the assays where we can find if antibodies or sera from vaccinated animals or humans are neutralizing. Neutralizing means that if they can block the entry of the virus into the cell. So if you can see on the pseudoparticle on the left, it has on the surface the SARS-CoV-2 proteins. So if there is an antibody present in the sample that binds to this protein, it can block the binding of this surface protein with the cellular receptor. And then we can see the decrease in the chemiluminescence readout in the assays. So, for example, in this graph, the gray bar is the bar of the positive control of the pseudoparticles infection, and then, for example, the blue bars represent a very neutralizing antibody with the higher dilution. We can see that the infection drops drastically, like about threefold, so that this antibody really can neutralize the virus. Then the next assay. Okay. We also wanted to check the influence of different subtypes of interferons on pseudoparticles entry. So interferons are the signaling molecules that are produced in the antiviral state, and they trigger expression of different antiviral proteins, also the IFITM proteins that inhibit the entry of viruses. So we were curious if different subtypes of interferon may be better or worse, and if we can observe an inhibition of entry. But here we didn't see actually any significant and dose-dependent inhibition of entry for those interferon. The interferons were kindly provided by Professor Catherine Ball from University of Edinburgh, and actually this is kind of in accordance of with the results of this scientific partner, because in their experiments it showed that these interferons act mainly on replication of genetic material, so somehow these experiments complement each other. And change. Okay. And also, we were still curious about IFITM proteins, how they act on SARS-CoV-2, but there were immediately some research papers showing this, and it was very interesting because for SARS-CoV-2, this is a little bit different than for other viruses. So researchers showed that when IFITM proteins are supplied to the cell and they are overexpressed, then they have antiviral function like for most of the viruses. But when they are expressed just endogenously, their function seems to be proviral. So because we were supplied with some anti-IFITM1 antibodies and sera from Dr. Borivoj Wojtesek from Rekamo Institute Research Center, we tried also to check if these antibodies can influence the entry of pseudoparticles to the cell. But also here we couldn't see, unfortunately, any dose-dependent influence on the entry. So, like, if I would like to sum up, I would say this pseudoparticle assay is very good for testing for neutralizing antibodies, because if we want the vaccine to be efficient, the neutralization should be quite high, and then we can see it very good on the neutralization level, infection level. But I think that because in this assay the standard deviations are pretty high and it's not easy to have like really very similar replicates, it is not so good for reagents that can give you very subtle differences between samples. So that is my conclusion. And now, yes, we use this system more for neutralization studies in our lab. Okay, so I would like to thank all the people that were supportive during this whole project. not just the COVID part. So first, I would like to really thank my boss from my former lab where this project took place, Professor Krystyna Bieńkowska-Szewczyk, and the students that were helping me with this project, and also all the colleagues, and also our partners at University of Edinburgh, Ghent University, and Recamo. Thank you very much.
Thank you very much. So are there any questions from the audience? If not, I have quick question. I understand your model could be suitable for pre-clinical screening of various drugs, and I feel that it could be suitable for example, antibodies or other biological drugs. But what about small molecules? Would it be suitable for this?
Yeah, like we can imagine there can be small molecules that inhibit entry, like there can be small molecules that bind to the virus or bind to the receptor of the cell. So then I think yes, yeah, but we must remember that it is just like this entry process of the viral infectious cycle. So I think it is mainly used as for antibodies. But of course, we can imagine some small molecules that could inhibit the receptor ligand binding.
Thank you very much. Okay, so if there are no other questions, so let's thank once again our speaker. Thank you very much. And our last but not least, Professor Marcin Drąg from Wrocław University of Science and Technology. So the floor is yours.
Okay, so good afternoon, everyone. And yeah, it's my great pleasure to talk about the, I think it's okay, yeah. Okay, it's my great pleasure to talk about the project, which I can tell you started a long time before even COVID started, that went from zero public interest to huge public interest. And basically, my lab is involved in proteases. So proteases in human, we have around 650 of them, and they control pretty much all metabolic pathways. And we are involved, and I'm involved personally more than 20 years in searching for the small molecules, inhibitors, substrates, and activity-based probes. But also, we don't work only on human viruses, we work also on other viruses. And we work on human proteins, on bacterial proteins, and all of the proteases related to them. And I will tell you today the story with the viral protease from SARS-CoV-2, the project which emerged from nothing. And some people say that I caused the pandemic to publish my results, but that was not the case. And I will tell you the whole story. So I can tell you that basically, if you look at COVID, it's run by proteases. It's run by 4 human proteases and by 2 viral proteases. If you look at the virus, there are 2 proteases which I will focus today on. It's MPRO and PLPRO, and I will tell you more why they are so important and why we think that they are the major reason for this bad COVID. And of course, we have more proteases. ACE2, which is known as the virus receptor, it's also a protease, it's angiotensin converting enzyme. And TMPRSS2, which cleaves the famous spike, is also a protease. So it cleaves the spike to make it better, to make better shape, basically, for the spike to insert the cell. And then, of course, we have cathepsin B and L, which are thought to be also involved in the COVID. And then it's like how they are involved and why they are important. So my story with SARS started more than 15 years ago, and I was States doing my postdoc. We're involved in the SARS-1. So I joined the Guy Salvesen lab in 2005, and we were working a little bit on looking for inhibitors for SARS. But at that time, there was a project with no interest because it was already after SARS. And 8,000 people sick, maybe around 800 people died. But then it's like we had lots of practice actually from SARS-1, how to work on SARS-2. And then if you look at SARS-2, basically, if it enters the cell, then it releases the viral RNA. And then this viral RNA is transformed by the human machinery into big polyprotein. So this big polyprotein then needs to be cleaved. Needs to be cleaved
by the proteases, and then there are two proteases, M-Pro and PL-Pro from the virus. So virus use its own proteases to cleave this protein, which is produced by the human machinery, and basically, if you stop this cleavage, you stop the replication. So when the Chinese, we're looking when Chinese will publish basically the genome of the SARS-2, and it happened on January 10th, and me and my collaborators, we look into the genome, and we immediately knew that basically we have the copy of the SARS-1 with these two proteases. So we had a lot of experience to immediately start the work. So why they are important? So we also immediately knew, long before even in Poland, people were freaking out, and in the world, people were freaking out about the COVID, that if you want to develop a drug, you have to develop the drug targeting either M-Pro or PL-Pro, and I will tell you why. So basically, if you stop the functioning, there will be no replication because there will be no virus copy produced, and if there will be no virus copy produced, then basically the virus will not develop. And of course, there were questions immediately about types of drug for COVID. So also knowing from SARS-1, we knew that there are three types of drugs basically you can develop. One is biological, which are antibodies, probably all of you heard about them. The problem with them is that they are not variant specific. So the virus mutates all the time, and usual antibodies are hitting the surface, which mutates a lot. So if you develop antibody towards one variant, maybe it will work with other variant, but it will be not pan-viral drug even for one type of the virus. So they were pretty much useless in long-term treatment of the COVID. Then you can have allosteric drugs, which are interacting somewhere on the surface of the protein, and then it's like they are useless too because of these mutations. So there is only one target you can basically target if you want to develop the pan-viral drug, which will work on all variants. It's active site directed. So active site directed is the place, basically active site of the enzymes, not only proteases, is the place usually in virus which doesn't mutate at all. Because if there will be mutation, then the virus will kill itself. So this is the only region which doesn't mutate at all. So let's start the story with the M-Pro. So M-Pro, if you look at this structure, it looks like a heart, and we call it also the heart of the virus. And it's very, very important protease. And basically, the story started a few years ago. So this is Rolf Hilgenfeldt. He's known for the SARS-1 work, and basically he published the crystal structure of the SARS-1 protease, and we collaborated with him for a long time on other viral proteases. And he was visiting Wrocław for a long time, and basically we were discussing about different projects, and he was telling me all the time that we had SARS, we have MERS, which is other coronavirus, so we will have the pandemic in the future, and he kind of tried to convince me that what about we develop the special library only for the coronaviruses, and maybe we can find the drug, because at that time, it was 3 years ago, there was no test for viruses, there was no drug for viruses, there was nothing, no vaccine. And basically, he kind of basically explained to me that if we have another coronavirus, it will be probably much worse than the ones we had before. So we decided actually to develop the library, but with no funding, okay? So we had no public funding for this project because no one was at the time interested at all. So we collaborate with pharma, and we heard about, sponsorship from pharma, and my lab is getting some money from pharma, and I can spend them on whatever I want. So one of the projects, basically, which we're doing in the lab with the hope that maybe we can find the application in the future, was developing special library for the viruses. And here is the library, and this library was basically synthesized in 2019, just before the pandemic, and I promised Rolf that he will come for Christmas to Poland and bring these two proteases from SARS-1 and MERS, and we will screen them. Maybe we'll find a leading structure which can be transformed into the drug. So what is the speciality of this library? If you look at these libraries, basically, they have you cannot see, but in the position 1, which I will show you just quickly faster, in this position here, it's glutamine. So there are no human proteases which accept human the glutamine amino acid. So this library is pretty much useless if you want to apply them for other projects using the human proteases. And if you develop this library, it's around 100,000 euros just to synthesize it. So there was no point, basically, for the project of zero public interest to synthesize this type of library, but because of this collaboration and extra money we had in the lab to spend on whatever, we decided to synthesize this library, and Rolf was about to bring us the enzymes, and at that time, he was asked to go basically to China. And then it's like, when when Rolf basically was in China in January, we started talking about getting, of course, SARS-2 protease to my lab, and he promised me that he will solve the crystal structure, come back from China, submit the paper, and the rest of the protease will come to my lab and we'll screen it. And we have the total monopoly in this type of libraries. They're only in my lab. So there were already many people asking us about what about we compare SARS-1, SARS-2, and tell what are the differences, if there are differences in the active site. And so Rolf, in the beginning of February, brought me basically MPRO from SARS-2. We screened it in 3 days. And we wrote this paper in another 2 days. And we're thinking, okay, we know what is the medical target. If you stop MPRO, you have 100% the drug for the COVID. And that was already February 2020. And we were thinking about what we should do with this data, and blocking this with patent or with whatever, where people were panicking about the COVID made no sense. So we decided to release it. And also we knew that we will not develop the drug by ourselves because we had no money, no resources, everything was closed. So this paper went viral from no interest to more than 100,000 views in few weeks. So basically all the big pharma immediately look at this data because they knew that MPRO is a really good medical target, and they wanted to use this information, and I will tell you how you can use this information. So here you can see that screening data where we use natural and unnatural amino acids, and more red, then more activity you have. So basically, if you have this red here, leucine, then you have high preference in one position. Then Pfizer, basically, they use this position, and in their first drug, which they put to the clinical trials in the September 2020, they had this leucine in P2 position, but then the drug was intravenous, so it made no sense to continue because they wanted a pill. And then it's like, if you look here in this position, so it's P3 position, it's crucial for the activity and selectivity of the virus, then you have these 2 amino acids in red. On the bottom, third leucine, and then on the top, D-tyrosine. So we published this data, and basically 1 year later, Pfizer introduced the second drug into clinical trials, which is now accepted as the drug and is used, and it's Paxlovid. So in P3 position, you can see over here. So it's here, basically, it's third leucine. So they use basically our scaffold to design the drug, and then this drug went immediately into clinical trials. So it's first drug containing a natural amino acid. So they basically beautifully validated the whole technology we have in my lab. And then it's like, this is the only drug accepted for now for the COVID. And of course, we published a nice paper in Nature Chemical Biology about this. We commercialized the substrate. So we signed a contract with the University of Technology. We use activity-based probe, basically. And I will tell you how. It's like people, they didn't want to believe that this drug binds so nicely. So basically, we solved the crystal structure, that it binds very nicely, and then there are unnatural amino acids in it. And with the help of Professor Wojciech Młynarski, we proved that actually we have the medical target completely confirmed, because the green picture is the COVID-positive patient, and this is the first image of the virus from the human trout, basically with the validated activity-based probe, which is the drug with the attached fluorophore. And then it's like we tested the molecule, which is our structure, comparing it to remdesivir, and it was really very successful and as good as remdesivir at that time with no cytotoxicity. And then we went into the second commercialization. We are selling with this company also this inhibitor. And I will very briefly tell you about the second protease, which is also really bad because it's PL-Pro. And we think that this is the major reason for the bad pandemic. And what is the reason why it's so bad? So basically, if you screen its activity, there are no mutations in the active site. So why is it worse than SARS-1, the protease? So we wanted to solve the crystal structure. We solved the crystal structure, and we were first, basically. And for a long time, we had the first small molecules with these inhibitors and the crystal structure published. But everyone was asking, what's the biological relevance of this protease, and basically why it can be so bad? So here you have the answer. So basically, in SARS-1, we knew from the research that it's besides the cleaving big protein, it's also silencing the cell. So if the virus gets inside, it cleaves the signal so the cell cannot defend itself, and if it cannot defend itself and then send the signal outside, kill me, then basically we will have another copies of the virus. So we found that actually this protease is even worse because it's more active on these human proteins. So it cleaves, it releases basically all this information, so the cell is basically blind. It's blind, cannot send the signal out, cannot adopt the signal from outside. So this way, virus can basically build lots of new copies, then destroy the cell, and then attack another cell. And we proved basically that this protease is one of the worst things which could happen, actually. And why this is happening? So this is the last slide, because we have so many mutations here. So if you look at this region, so you have the catalytic site region, no mutation at all, because the virus will kill itself. But then you have tens, and now even hundreds of mutations on outside. So all these mutations make all the antibodies and all the other, let's say, the drugs which don't interact with the active site totally useless, while basically you can use this for the mapping of the drug in the active site, but also it shows that basically it's so active on this human substrate, so make totally the cell blind. So with this, basically, we could prove that proteases are excellent drug targets, both of the proteases. Their binding sites are identical with SARS-1. So, and then there are more and more data coming out. We just submitted another very big story yesterday showing that there is no mutation in the active site of this protease in all possible really dangerous variants. And we need the drugs for the next coronaviruses because vaccine will not work. We have the vaccines, but then to develop new vaccine, it will take a couple of months probably if we have the new pandemic with the new virus. So we need a general antiviral drug which will help us basically with this. And then it's like, of course, there is a queen of this project, is Viola. She was working for many days, 24 hours a day, to screen all these libraries, to generate all this data, but she finished with 4 first author papers in top journal in 1 year. So, and then, of course, we had many, many collaborators working with us, so it was really very nice and big projects, and thank you, Foundation for Polyscience, for sponsoring our research, and thank you very much.
Thank you so much. Lots of wonderful information from the scratch, and the path to commercialization is waiting for you, Marcin. So are there any questions from the audience? I really would like to ask about this vaccination and drugs, what is better or no, but I'm afraid we don't have plenty time to discuss it. Could you quickly?
So now with vaccines, if we have vaccines and we have the new virus, the vaccine will not work, because we see that with new variants, the vaccines are less and less effective. So we need a drug, and that was the major dogma for SARS-1 when we were working, because all the companies, they wanted to have antiviral drug. If we have the antiviral drug, and we can now see that there are no mutations in the active site, so with no mutations in the active site, and probably with next coronaviruses, it will be the same. So Pfizer, they published their data on all known coronaviruses, and this drug works on all known coronaviruses. So it's not like vaccine that it works on SARS-2 only. The drugs they have developed on this technology basically works on all possible and known coronaviruses so far. So probably we will need a drug to stop the pandemic, because this coronavirus, many of you say is really bad. It's very nice, because the mortality rate is very low. It's like a few percent, maybe 2-3%, comparing to MERS, which we have now, where the mortality rate is around 45 to 50% even, and maybe we can have another virus with mortality rate of 50%. So before we develop the vaccine, then many, many lives will be gone.
Yeah, okay, thank you very much. So let's thank again. And before we thank all the speakers, I would like to say that there are no doubts that due to pandemic, development of vaccines, drugs, and diagnostic tests has been massively accelerated. But what is also important, and I would like to underline, that due to pandemic, we also learned a lot about ourselves. So thank you very much, all the speakers, for your contribution to this library I mentioned at the beginning of this session. And let's thank once again our speakers that they were ready to share their data and conclusions with us.
Thank you, Radek, and also thank you very much to all the speakers for sharing this information from so different area of inquiry. I'd also like to thank you, our online participants. It was our last session today. We'll meet here again tomorrow at 10 o'clock. And goodbye. The streaming is now off, and as our conference started on more than 8 years ago, I'm pretty sure we are nourished with new knowledge and